Welcome to my presentation of Chapter 24, the Urinary System. So the main structure, the main organ in the urinary system is the kidney, which you have two of. The job of the kidneys is to filter the blood and make urine. And the word renal means related to the kidney. So anytime you see the word renal, it means that they're talking about the kidney. Apart from the kidneys, most of this presentation will actually be about the kidneys, but apart from the kidneys, you have the urinary tract, which is basically responsible for taking urine from the kidneys and expelling it from the body. Within the urinary tract, you have three different organs. You have the ureters, two of them, one urinary bladder, and one urethra. So on this diagram, you have the kidneys here. The adrenal glands are sitting on top of the kidneys. Then you have the ureters leading from the kidneys to the bladder, which technically would be called the urinary bladder. And then you have the urethra leading from the bladder to the outside of the body. In women, the urethra is short like this. In men, it is longer because it goes through the penis, which is nice for men because it means that they don't get as many urinary tract infections as women do. So in general, the functions of the urinary system are done by the kidneys. Most of them are related to filtering water and solutes from the blood to make urine, but you also have hormones that are released from the kidneys. The kidneys release renin, which is responsible for activating the RAAS, and we'll go into the details on the RAAS later in this presentation. And you also have the kidneys releasing EPO or erythropoietin for erythropoiesis. So one of the main functions of the urinary system is excretion. You have two basic ways of excreting waste products from your body, defecation and urination. So the kidneys are in charge of urination, or making the urine in the first place. You have a lot of different wastes actually that would be removed by the kidneys. Anything that's in the blood that's water soluble can end up being excreted by the kidneys into the urine. But the prominent wastes would be urea and ammonium, which are both types of nitrogenous waste, which means that they have nitrogen in them. So those things would come mostly from breakdown of protein. They can also come from other sources. If they build up in the blood, it's quite dangerous. They can alter the pH of the blood and alter the pH of the whole body, which would eventually be fatal. So those things are excreted in the urine. Also anything else that's water soluble that is toxic. And I'll just mention the fancy name for urination is micturition. I guess urination wasn't fancy enough, so they have micturition, which means basically peeing. Another function of the urinary system is fluid homeostasis. So the kidneys are able to control how much water ends up in the urine, which is one of the main ways that your body controls how much fluid it has. Between losing water in urine and gaining water by drinking it, those are the two main ways that you're getting water in or out of your body. And so those two processes are balanced to maintain the proper osmolarity of your blood, which would be the proper concentration of solutes in your blood. The urinary system also helps to maintain the blood pressure because it's related to excreting urine, which reduces the blood volume. And when you reduce blood volume, then you reduce the blood pressure. But in addition to that, the kidneys release renin, which activates the RAAS. If you have renin being released, that means that you're going to have angiotensin II and aldosterone being activated / released. And both of those hormones affect the blood pressure by increasing it. And we'll go into the details on that later on. Then next you have electrolyte homeostasis. Electrolytes are basically like ions, important ions that you need for your body to function correctly. So the urinary system and the kidneys are in charge of making sure that you have the correct amount of each electrolyte in your blood, and any extra is going to be excreted in the urine. So some of the most prominent ions that are controlled that way would be sodium, chloride, and potassium, but you also have others that are a little less common, like magnesium, that are controlled that way by the kidneys. Next you have acid-base homeostasis. So the kidneys are able to control how much hydrogen ions and how much bicarbonate is excreted into the urine. And by doing that, they can affect the pH of the blood. So if you have too low of a pH, that would be acidic. That would mean you have too many hydrogen ions. So one way you could fix that is by the kidneys excreting more hydrogen ions in the urine. Then bicarbonate is part of the body's buffer system. And it also affects pH. In addition to controlling how much bicarbonate is excreted in the urine, the kidneys can actually make some bicarbonate. So if the pH of the blood is dropping too low, then the kidneys would start retaining bicarbonate and they would start making some extra bicarbonate. Then the bicarbonate is going to combine with some of those free hydrogen ions, which will raise the pH. Then finally, the kidney functions in regulating erythropoiesis because it is the source of EPO, the hormone that triggers the bone marrow to make red blood cells. Those are the main kidney functions. There's also some other ones, but we won't talk about them. So we'll go through the other parts of the urinary system now beyond the kidneys. First, the ureters. There are 3 layers in the wall of the ureters. You have the mucosa, the muscularis, and the adventitia. That's from deep to superficial. So the mucosa is the innermost layer, and the adventitia is the outermost. The mucosa is composed of transitional epithelium, and there's also a small submucosa beneath it made of loose connective tissue. Transitional epithelium, as you may remember from ANP1, is a special type of epithelium. It's stratified, and the cells in the top layer have domed shapes, so that if the tissue stretches, those cells will just flatten out, and it allows the tissue to stretch without damaging the epithelium. The only real place in the body where you find transitional epithelium is in the urinary system, and it allows all the organs of the urinary system to expand to accommodate urine. Then after the mucosa, you have the muscularis, which is a smooth muscle layer. The smooth muscle in the muscularis contracts in a coordinated pattern called peristalsis, which we would have talked about when we talked about the digestive system, but in peristalsis you have a ring of smooth muscle around the ureter contracting, and then that ring basically slides down the ureter. So the smooth muscle in one location will contract and then release as the smooth muscle just below it is contracting. That way urine is pushed through the ureter down from the kidneys to the bladder. Then you have the adventitia made of dense irregular connective tissue, which is just preventing the ureters from overstretching and rupturing. So the ureters drain urine down into the bladder. They enter the bladder on kind of the inferior aspect, so the underside of the bladder, and they have this diagonal entry. So this tube going in here would actually still be considered the ureter, and then it finally opens into the bladder here. And this diagonal path that the ureters take through the bladder wall is important because it prevents urine from going back up into the ureters once it's reached the bladder. So as your bladder fills with urine, this is going to get pushed shut here. The urine will be pushing on the walls of the bladder, and that squeezes the ureter shut where it's going through the bladder wall, and so nothing can get back in to the ureters. So most people will have kind of a long stretch of the ureter that is tunneling diagonally through the bladder wall, but some people are born with a shorter tunnel through the wall, or with just a straight tunnel going straight through the wall that doesn't have a diagonal angle to it. People who have these conditions are at risk for urine going back into the ureters out of the bladder and backing up even into the kidneys, which could be dangerous because there's not enough of the wall to be squeezed shut. Next you have the urinary bladder, which has 3 layers in its walls. First you have the mucosa again, that's the innermost or deep layer, then the detrusor muscle is the middle layer, and then the adventitia is the superficial or outermost layer. The mucosa in the bladder is again transitional epithelium with a small layer of loose connective tissue, that's the submucosa, that would be supplying the epithelium with a blood supply. And again, that epithelium can stretch. It is secreting mucus. Actually, the epithelium in the ureter secretes mucus as well. That's why it's called mucosa. And that mucus is important because the urine has a lot of toxic products in it. And so if you had cells that are continually exposed to the urine directly, they would be damaged by it. So the mucus is protecting the walls of the bladder from the urine, just similar to the way that mucus in your stomach protects the walls of the stomach from the stomach acid. Then most of the bladder wall is composed of the detrusor muscle, which is a specialized smooth muscle. You actually have 3 different layers of that muscle, and the fibers are running in different directions in each layer. Right where the urethra is opening from the bladder, you have kind of a specialized part of the detrusor muscle that goes around in a ring, and that forms the internal urethral sphincter. So you actually have 2 sphincters that are controlling the exit of urine from the bladder through the urethra. You have the internal one formed by the detrusor muscle there, and then you have the external one down here. And then finally, at the most superficial part of the wall is the adventitia, which in this case is composed of loose connective tissue. Usually the adventitia is made of dense irregular connective tissue for strength. In the case of the bladder, it's loose connective tissue. To be honest with you, I'm not sure why that is. If I have to take a guess, I would say that maybe the bladder needs to be able to stretch more than what dense irregular connective tissue would allow. Next, the urethra. The 3 layers of the urethra wall are the same as the 3 layers you have in the ureters. So it's again the mucosa, the muscularis, and the adventitia. You have 2 sphincters in the urethra. First, you have the internal urethral sphincter, made of a specialized part of the detrusor muscle of the bladder. And then you also have the external urethral sphincter, which is formed by the levator ani muscle, which is shown right here highlighted in green. So the levator ani muscle is forming most of the pelvic floor, kind of just filling in the gap between the coxal bones there, so that your bits don't fall out the bottom. The internal urethral sphincter is composed of smooth muscle, so you do not control that consciously. It will open, it will relax whenever the bladder is full of urine, and you have stretched receptors in the walls of the bladder that can sense when it's full. The external urethral sphincter is under your voluntary control, so if you're holding it in, you're using the external urethral sphincter to do that. The internal one is probably already open because it's time. The male urethra is different from the female urethra. This image here is of a female urethra. It just opens directly into the vagina, and it's very short. The male urethra is much longer. You can see it starting here. This would be the bladder. Oh, sorry. This is the bladder here. And then here you have the urethra traveling down to the opening. So it would be going through the prostate gland. It actually has 3 different parts. So you have the prostatic urethra, where it's traveling through the prostate gland. You have the membranous urethra, where it's traveling through the levator ani muscle. Then you have the spongy urethra, where it's traveling through the penis. So it's much longer than the urethra in women, which means that if bacteria are going to come up here, they have quite a long way to go before they can reach the bladder, and that makes men much more protected from bladder infections than women and UTIs than women are. In women, as you can see, it's a very short distance for bacteria to travel to reach the bladder. Also in men, the urethra is not just carrying urine, it's also carrying semen for ejaculation. So the prostate gland, the testes are responsible for making the semen in the first place, and the prostate gland modifies the semen. So that semen would be coming through this tube here, going through the prostate, and then that merges with the urethra so that it ends up coming out the urethra. I'll just briefly mention urinalysis right here. Urinalysis means studying the urine, and it is done to gain information about a person's medical conditions and their health state. You can find out a lot about a person's state of health by looking at their urine, especially, obviously, if they have something wrong with their kidneys or with their urinary system. So the first thing you can look at, which actually everyone can look at in their urine, is the color. It should be pale yellow. If it doesn't have any color in it at all, that means that you are overhydrated, and your body is producing very dilute urine to get rid of some of that extra water. If your urine is dark yellow or even a different color, like so dark it's like orangey or something, that means that usually means that you're dehydrated, and so your body is retaining all the water that it can manage to hang on to, and producing a very concentrated urine. So that's a good way to gauge your level of hydration. That's probably the best way to gauge how hydrated you are and whether you need to drink more or less water. Your goal is always pale yellow urine. The color there is actually coming from the compound urochrome, which has a yellow color. Urochrome, as you may remember, is produced during the degradation of hemoglobin. So when your red blood cells get worn out and are destroyed in the spleen, the hemoglobin, the heme part of it actually, the heme part of the hemoglobin is degraded to form several different products, one of which is urochrome, which is excreted in the urine and gives it that yellow color. The intensity of the yellow color is determined by how much water you have in the urine in general. And how much water you have in the urine should reflect your hydration level. It's also possible to have other colors in the urine, so certain drugs and certain foods can give a color in the urine. Also, if you have blood in the urine, that will obviously give it a color, and you know, that's not good. Next, the urine should be translucent, which means you should be able to see light coming through it. If the urine is cloudy, usually that means you have an infection, a urinary tract infection, and there's bacteria in the urine making it cloudy. It could also mean that there's too much protein in your urine. Usually there should be very little protein in the urine. But in general, it usually means an infection. Actually, if you leave urine sitting out for too long, it'll start growing bacteria and become cloudy on its own. So in order to assess the translucency, you need to kind of look at it right away. Next, you have odor. That should be not too much of an odor. Again, if you leave the urine sitting, then bacteria are going to start eating it and producing waste products, which have an odor. So to assess the odor of your urine, you have to do it quickly. The odor can be changed if you have an infection, so if there's bacteria in there. Certain foods can also change the odor, and if you have diabetes or high glucose, if you're hyperglycemic and you have too much glucose in your blood, then glucose will end up being excreted in the urine, which gives it a sweet smell. So if your urine smells sweet, that's a good sign that you've been eating too much sugar. You need to cool it with that candy. You can also look at the pH of the urine. It should be around 6. The specific gravity means it means how many solutes are in the urine compared to how many solutes are in deionized water, which would be like pure water. So in general, the urine should be around 1.001 to 1.035 specific gravity. So if you have dilute urine, that should be almost just water, and that would be a specific gravity of 1.001, almost as concentrated just as water. If you have very concentrated urine, the specific gravity would be up around 1.035. If you have more than that, then that means that there's too many solutes in the urine. Something's going wrong with the way that it's being formed. You shouldn't have many cells in the urine. The urine itself should not have any cells at all as it's being formed, like 0 cells at all. But as the urine is traveling through the urinary tract, it can pick up some cells. So you can have some epithelial cells that are dying and shedding off the walls of the urinary tract. Also, I mean, sometimes you do have like a red blood cell or a white blood cell make its way into the urine, even though that really shouldn't be happening. But if it happens just a little bit, then that's not necessarily abnormal. But if you have a significant amount of cells in the urine, then that means that something is going wrong. Particularly if you have a lot of red blood cells or white blood cells. That can mean, if it's a lot of white blood cells, that can mean you have an infection and they're being attracted to the kidneys. If you have a lot of either type of cell, that could also mean that there's something wrong with the filtration in the kidneys themselves. They're not properly filtering the blood and they're letting big things get through that shouldn't get through. Again, you have to be careful when you're trying to look at the cells in the urine. You have to be careful to get what's called a clean catch, which would mean urine that is not contaminated by its passage through the urinary tract, which to get a really clean catch is actually pretty hard to do. For a perfectly clean catch, you actually need to use a catheter to collect it directly from further up in the urinary system. In addition to all those basic things, you can analyze the urine for specific solutes like glucose or ketones or amino acids, just different things. Anything you're interested in looking at, you can analyze the urine to see if it's there. That'll give you information about specific conditions. So now we'll go through the renal anatomy. Actually, for the rest of this presentation, we're really going to be focusing on the kidneys themselves, the anatomy of the kidneys, and then the physiology. So the external kidney anatomy, you have 3 membranes around the kidneys on the external surface of the kidneys, made of connective tissue. So the most superficial of those is the renal fascia, then you have the adipose capsule, and then the renal capsule would be the deepest, so that would be the one that is right up directly against the surface of the kidneys. The renal fascia is composed of dense irregular connective tissue. Let's see. In this diagram, the renal fascia is this pale line kind of on the outside. And as you see, it's kind of far away from the kidneys themselves, especially anteriorly. That renal fascia is connecting the kidneys to other structures in the abdominal cavity so that they stay in place. Then you have the adipose capsule underneath that, made of adipose tissue. So that would be all of this yellow stuff around the kidneys. The role of that adipose capsule is usually to absorb shock, just to cushion the kidneys against shock and impacts. But if you are starving, then your body will start eating that adipose capsule, which would cause the kidneys to droop within the abdomen. And then the innermost membrane is the renal capsule, also made of dense irregular connective tissue, and that would be just right up against the kidneys themselves. You can actually see it in some of the dissection specimens. It's pretty thin, so it's kind of hard to see, but sometimes you can see it. So the renal sinus, oh sorry, the hilum. So the hilum is the point on the kidneys where things are entering and exiting. That would be right here on the kidneys. So you have arteries going in, veins coming out, the ureter coming out, and nerves going in and out through the hilum. Then the hilum is opening up into the renal sinus, which is like, it's basically all of the space inside of the kidneys. So you have some of the internal structures of the kidneys housed inside the renal sinus. Is not actually labeled on this diagram, but it would be including from the hilum all the way up here all of that yellow stuff there, that would be the renal sinus. And then on this side, all of this yellow stuff starting at the hilum and then going into the kidneys. So the renal sinus is kind of the space in there where you have your arteries and veins and nerves passing through it. The ureter would be opening up off of it into the hilum. And then your internal structures that are in the sinus would be the renal pelvis and the major and minor calyces, which we're going to talk about directly. The renal sinus can be kind of difficult to distinguish from the renal pelvis, so I won't ask you to do that. So internally to the kidneys, we have the renal cortex. That is the outermost layer, or the most superficial layer. So actually in this diagram, this red stuff would be the renal capsule, and you can see there's the thin layer there of the renal capsule, and then the kind of lighter stuff, the lighter red beneath it, that is the renal cortex. Then the middle layer is the renal medulla, and the innermost layer is the renal pelvis, which is also part of the renal sinus. Within the renal cortex, you have nephrons, which are the functional unit that is actually forming the urine. Then you have these extensions of the cortex down into the medulla. Those are called the renal columns. Those columns are containing blood vessels. So you can kind of see the little dashes, I guess, of blue and red. Those are blood vessels traveling up through the renal columns. And in this image, you can see it better. So you have this artery, this renal artery going into the kidney. It branches and it sends smaller arteries up the renal columns to get to the proper part of the cortex. And then the veins would be coming back down through those renal columns to the renal vein. Within the renal medulla, you also have part of the nephrons that are making urine. So you're actually kind of forming the urine in the first place in the cortex and then adjusting it in the medulla as far as how much water it has and how much of different solutes like electrolytes it has. And these kind of, well, they sort of look like pyramids. These little triangles kind of darker brown are the renal pyramids, which is the main kind of structure that you find in the renal medulla. And here you have them again. So you have a pyramid, that's the renal medulla. Then you have a column, that's actually renal cortex. Then another pyramid for the medulla. Then a column for the cortex. Blood vessels going through the columns, and urine being adjusted in what do you call it? The pyramids. And then the urine, as it's formed, would be draining from these renal pyramids into the renal pelvis, into this yellow area, into this yellow space. You have kind of right here where the renal pyramid is first draining into the pelvis. There you have what's called a minor calyx. The calyx means cup. So it's a cup that is holding urine and passing it down. And then as a couple of these minor calyces merge together, you'll have a major calyx formed. So this would be another major calyx with 3 little minor calyces coming off of it. And then the renal pelvis proper would begin just beneath those major calyces. All of that, the renal pelvis, the major calyces, and the minor calyces, all of that is part of the renal sinus. And here you can actually see the renal sinus labeled on there. So this whole space inside of the kidney is the renal sinus, and then we're dividing different parts of it into the renal pelvis, the minor calyces, and the major calyces. If you zoom in on the kidneys further, you'll find the nephrons. The nephron is the functional unit of the kidney. Most organs actually are composed of functional units, which would be kind of the minimum structure necessary to carry out the task of of that organ. So for instance, in bones, the functional unit would be the osteon. And if you have a lot of osteons together, then you're going to get a strong bone. In the kidney, the nephron is the functional unit. Most of the kidney is made of nephrons. The nephrons are filtering out the urine in the first place from the blood and then adjusting its contents. Although actually, technically, once before the urine is fully formed, when it's still being filtered and adjusted, we technically don't call it urine yet. We call it filtrate instead, to reflect that it's not finished. So within the kidney, you would find the nephrons mainly in the cortex of the kidney, but then they also descend into the medulla. You have a couple different types, so some of them descend further into the medulla than others. And you have two basic parts of the nephron. First, you have the renal corpuscle, which is this little ball. Then you have the renal tubule, which would be this long, squirming tube coming off the nephron. And then this big, long structure here is a collecting duct. So that's not technically part of the nephron, and you can see that you have multiple nephrons that have their tubules draining into this collecting duct. So in the renal corpuscle, that's where you're first forming the filtrate. You're actually filtering the blood to get the filtrate, and then the filtrate travels through these tubules. As it travels, its contents are adjusted as far as how much water there is and how much of different solutes there is in it. Then it reaches the collecting duct, drains into the collecting duct, and travels down to the minor calyx. By the time it reaches the minor calyx, it's considered now urine, not filtrate anymore. And here you have an image of the nephron that has the blood vessels also shown. So you would actually have, here you have an artery. This would be traveling across the top of a renal pyramid. So this artery here would actually be traveling across the top of maybe this pyramid right here. Then it's going to send up a smaller branch, and then another branch, which we call the afferent arteriole, which is going to go into the renal corpuscle. In the renal corpuscle, you have a capillary bed, which is called the glomerulus capillary bed. Then you have an efferent arteriole that comes out of that glomerulus capillary bed, and then it enters into another capillary bed called the peritubular capillary bed, and that peritubular capillary bed stretches across the entire network of tubules. Well, not really network, but the whole length of the winding tubule. Those capillaries are very important. The glomerulus capillary bed is where blood is initially filtered to get the filtrate that will eventually become the urine. And then the peritubular capillaries are important for adjusting the contents of the urine. So you can take more things out of the blood and put them into the filtrate, and you can also take some stuff from the filtrate and put it back into the blood in those peritubular capillaries. Also, the glomerulus capillary bed, I should mention, is an unusual capillary bed. Usually you would have an artery, then a capillary bed, then a vein, or actually an arteriole, then a capillary bed, then a venule. But in this case, you have an arteriole, a capillary bed, an arteriole, and then another capillary bed, and then a venule. So if we zoom in on the renal corpuscle within the nephron, we find that it has two parts. First, you have the glomerulus capillary bed. Then you have the glomerular capsule. So this entire structure here is the renal corpuscle. The glomerulus is in here. It's actually coated by the glomerular capsule right now. If you peel that away, you would just have the red capillaries in there. Then this structure, like a ball around it, is the glomerular capsule. You actually have two layers of the glomerular capsule. The outside layer here is the parietal glomerular capsule. That's made of simple squamous tissue. Then you have the inner layer, which is also the visceral glomerular capsule, and that is composed of special cells called podocytes. So you can see there's a podocyte. And the podocytes have these little processes that come off of them. So you can see all these tiny yellow lines are those processes of the podocytes that kind of interweave with each other to form little slits called filtration slits. So if we look at the glomerulus, the capillary bed, the glomerulus capillary bed, the capillaries are fenestrated capillaries, which, as you remember, means that they have the little holes in them and little gaps between them, so that stuff can get out. Big things wouldn't come out, but like really big things wouldn't come out, but a lot of stuff can get out. Any type of solute would be able to get out through those fenestrated capillaries, and then it meets the podocytes in the visceral glomerular capsule, and would be filtered through those little filtration slits. So here you have an electron micrograph view of the glomerulus with the visceral glomerular capsule on top of it. So beneath here in red, showing through, those are the glomerular capillaries. Then this is a podocyte, and this is a podocyte, and these little lines coming off of them are the podocyte processes. And then you can see even smaller little lines coming off of those, which are also the podocyte processes that interweave with each other to make the filtration slits. So you have teensy little spaces, really, really small spaces between these processes that are the filtration slits. So you would have blood passing through the glomerular capillaries, and as it goes through, it's going to be filtered through the fenestrations in the capillaries and through these little filtration slits. And then you're going to have water and solutes from the blood ending up in the space between the visceral and parietal glomerular capsule. That space can be called the capsular space, but it's also just called the lumen. Lumen is just a general word for the inside of any hollow organ. So the insides of the blood vessels are called the lumen of the blood vessels, and the inside of the glomerular capsule is called the lumen of that capsule. So I'm just going to call it the lumen because, you know, it's just one less vocab word, you guys. So you would have blood filtering out through these glomerular capsules and the glomerular capillaries and the filtration slits, and that makes filtrate filling up the lumen of the glomerular capsule, and then that would enter into the renal tubules, leaving the corpuscle and going into the tubules. So now zooming in on the tubules, you have 3 different segments of each tubule. You have the proximal tubule, which would be the part that is closest to the corpuscle, and as you can see, it's winding around. So it's sometimes called the convoluted proximal tubule. Next you have the nephron loop, which is this loop that goes down and comes back up. That's also called the loop of Henle. A lot of kind of older sources will just call it the loop of Henle. Then you have the distal tubule, which also starts winding around and empties out onto the collecting duct. In the proximal tubule, it is lined with simple cuboidal epithelium, and you have microvilli on the surface of the cell, the apical surface that faces into the lumen of the tubule. So there's your microvilli. Right, here's your simple cuboidal epithelium, and then the microvilli, and then the interior of that tubule. The microvilli increase the surface area so that you can have a lot of absorption or transport occurring across the cell membrane there in the proximal tubule. Then you get into the nephron loop. That's divided into the descending limb and the ascending limb. The descending limb goes down into the renal medulla, into the pyramids, and then the ascending limb comes back up to the renal cortex. The descending limb is mostly lined with simple squamous. Actually, you can see this thicker part up here, that's lined with simple cuboidal, and then most of it is lined with simple squamous. And then the ascending limb, most of it is lined with simple cuboidal, but also at the bottom you have some that's lined with simple squamous. So technically, this would be divided further. This is the thick descending limb, this is the thin descending limb, then you have the thin ascending limb and the thick ascending limb, but we won't get into all of that. Then the distal tubule is lined with simple cuboidal epithelium. Only the proximal tubule has simple cuboidal cells that have microvilli. Everywhere else in the renal tubules where you have simple cuboidal, it does not have the microvilli. Then you also have one part of the distal tubule that's kind of more specialized and participates in what's called the juxtaglomerular apparatus, and that is shown right here. So here you have your renal corpuscle. This would be the afferent arteriole going into the glomerular capillaries, and then coming out as the efferent arteriole. Afferent means going to, as in to. So to this glomerulus capillary bed. Then efferent means going from, e like exit. So the efferent is going away from the glomerulus capillary bed. And then right there you have part of the distal tubule that loops around to get real close to the afferent and efferent arterioles. Up here you can see it again. These are those two arterioles, and then here you have the distal tubule getting close to them. So this right here is your juxtaglomerular apparatus. Juxtaglomerular because it's next to the glomerulus. In green, you have special cells that are lining that distal tubule. Those are called macula densa cells. And then you have those cells connecting to juxtaglomerular cells. In orange and in blue here, you have the juxtaglomerular cells. Those cells, well, at least the blue ones, are actually smooth muscle cells that are part of the smooth muscle layer in the arterial wall. The point of that juxtaglomerular apparatus is to help the kidneys control the rate at which they're filtering urine, while filtering the blood to make filtrate, and so we're going to come back to that later on. Then you have, as I had mentioned, two different types of nephrons. Most of the nephrons are cortical nephrons, and then you also have some that are juxtamedullary nephrons. So these would be the cortical nephrons. Most of them is in the renal cortex, which is why they're called cortical nephrons. You just have a teensy bit of the nephron loop that does descend down into the renal medulla. The nephron loop also, as you notice, isn't very long. In the juxtamedullary nephron, the nephron loop is way longer, and the nephron itself is just closer to the renal medulla. So the proximal and distal tubules and well, actually this would be the proximal and this would be the distal. Proximal and distal tubules and then the corpuscle are all at the very bottom of the renal cortex, and then the nephron loop is almost entirely within the renal medulla and goes very deep into it. Another difference between these two types of nephrons is the blood supply. So in your cortical nephrons, you have the glomerulus capillary bed, then the peritubular capillary beds going all around the tubule part of the nephron. In the juxtamedullary nephrons, you have the glomerulus capillary bed and the peritubular capillary bed, but the peritubular capillary bed is only going around the proximal and distal tubules. The nephron loop has a totally different capillary bed around it called the vasa recta, and that's important later on. The juxtamedullary nephrons are very important for controlling the water content and the salt content of the urine. They're very important for allowing you to make concentrated or dilute urine, and we're going to come back to that later. So next we're going to start talking about the renal physiology, or how the kidneys work. And the basic thing they're doing is making urine, so we'll be talking about urine formation. You have three basic processes that occur during urine formation: glomerular filtration, tubular reabsorption, and tubular secretion. Glomerular filtration occurs in the corpuscle. That's basically your initial filtering of water and solutes out from the blood. So all that stuff would be coming out from the blood, going through the glomerular capillaries, and it ends up in the lumen of the glomerular capsule. Then it's going to enter the tubules. Then the filtrate will enter the tubules. So glomerular filtration is just the process of making that filtrate in the first place in the corpuscle. Then in the tubules, that's where you have tubular reabsorption and tubular secretion. Tubular reabsorption means taking some of that stuff that's in the filtrate and returning it to the blood. So in reabsorption with the R's, you would have solutes and water leaving the filtrate and going into the peritubular capillaries to join back with the blood. You have a lot of that in the proximal tubule, also in the nephron loop. Well, you have it actually everywhere. So it's in the proximal tubule, in the nephron loop, in the distal tubule, and even a little bit of it in the collecting duct itself. But most of the reabsorption is occurring either in the proximal tubule or in the nephron loop. Then finally you have tubular secretion, which means taking stuff from the blood and putting it into the filtrate in the tubules rather than in the corpuscle. So as your blood is passing through the peritubular capillaries, you can have different solutes, generally toxic wastes, transported out of the blood and into the kidney tubules, into the tubule lumen, into the filtrate. That would be mainly occurring in the proximal and distal tubules, and also some in the collecting duct. So glomerular filtration is the basic process of forming the filtrate in the first place, and then through tubular reabsorption and tubular secretion, you are adjusting the contents of the filtrate to be appropriate. That way you can make sure that you have the right amount of water in the blood, the right amount of different solutes in the blood, the right pH in the blood. So the tubular reabsorption and secretion are really important for making sure that the contents of the urine is just right. So we're going to go through all these processes in turn, but they're basically contributing to the water homeostasis that's maintained by the kidneys, the waste excretion that's done by the kidneys, the electrolyte homeostasis maintained by the kidneys, the acid-base homeostasis maintained by the kidneys, and the blood pressure homeostasis maintained by the kidneys. So urine formation is accounting for the bulk of the functions of the kidneys. So now we'll go through the process of glomerular filtration in a lot more detail. So it all starts with the filtration membrane, which we briefly touched on before. That is the membrane through which blood is filtered to form the filtrate in the corpuscle. There are 3 layers to the filtration membrane. You have the endothelium of the glomerular capillaries. Then you have the basement membrane of those capillaries. And finally, you have the podocytes, which is the visceral glomerular capsule. So the glomerular capillary endothelium is it's fenestrated, where the glomerular capillaries are fenestrated capillaries. So they have little gaps between the cells here and little holes in the cells. And large solutes can pass through those. So any large molecule that is in the plasma can get through the glomerular capillary endothelium, but the cells in the blood cannot. So the leukocytes, the red blood cells, and the platelets are much too big to pass through those fenestrations in general. The basement membrane is technically also part of that epithelium that is lining the capillary or forming the wall of the capillary. And it filters more finely than the capillary endothelial cells do themselves. The basement membrane has a lot of collagen fibers in it that are running every which way, and they form a sort of net, and that would be filtering out most of the large solutes. So proteins would be the largest solutes that you find in the plasma. Most of them are too large to get through the net of the basement membrane. All of your small solutes can get through your ions, small nutrients like your glucose and amino acids, Yeah, anything small can get through, but the really big proteins cannot. Also, some tiny proteins can get through there. Some little peptides will get through. The other thing about those collagen fibers is that they have a negative charge, which means that some of the things that are small that have a negative charge also would be repelled by that charge. So a positively charged ion has an easier time getting through the basement membrane than what a negatively charged ion would have. Then finally you have the podocytes, which are forming the visceral glomerular capsule. So here would be the cell body of a podocyte, and then they send out their little processes, and each process sends out more little processes, and those interweave to form the filtration slits, the little tiny little gaps between processes of the podocytes. So if you zoom in, you see all these little triangles are the cross-section of the podocyte processes, and you would have one belonging to one podocyte, and this belongs to that podocyte as well, and this one, but then the ones in between there belong to a different podocyte, and they're interwoven. And you just have these teensy little gaps between them. The gaps between them are very, very small. So only things that are very small can pass through those. So all of your small proteins would be excluded, or almost all of your small proteins would be excluded by those filtration slits. Only the smallest solutes will make it through, and water itself. The filtrate would be formed of everything that did make it through the filtration membrane from the glomerular capillary bed. It contains water, of course, and then dissolved in the water you have different ions, which in this case we're usually using interchangeably with electrolytes. And then in addition to that you have glucose, you have the teensy-weensiest proteins, the little peptides. Some peptides can make it through. You have amino acids, which would be linked together to make the proteins. Then you have waste products. Most of the waste products would be nitrogenous waste. Those are wastes that contain nitrogen atoms. The most prominent one is urea, and some of that urea is used to make ammonium. Ammonium is NH4+, so that's an ion. Urea and ammonium are generated from the catabolism of protein. So anytime you're breaking down protein for whatever purpose, if you're using it for energy or even if it's just an old protein that you're recycling, you will end up either reusing those amino acids to make a new protein or getting rid of them. And if you're going to get rid of them, you're going to end up with nitrogenous waste, urea, and ammonium. Those things are toxic. If you don't do anything with them, they will build up and be quite toxic, so you have to get rid of them. You also have uric acid. That's another nitrogenous waste. This one comes from the catabolism of nucleic acids. Nucleic acids would be the building blocks, nucleotides would be the building blocks for your nucleic acids, which are DNA and RNA. Anytime you're breaking down a nucleotide, it contains a nitrogenous base that has nitrogen in it, and if you're breaking that down to get rid of it, you're going to generate uric acid. Uric acid is different from urea because it is not as water soluble as urea is. That means that you cannot filter quite as much of it out into the filtrate as you can with urea. So it's kind of more dangerous to be accumulating uric acid because it's harder to get rid of in the urine. And actually, if you are having trouble with your kidneys so that they're not filtering things quite properly or doing it quite enough, or if you're having quite a lot of nucleic acid catabolism, then you're going to end up with uric acid that cannot be eliminated, and that is actually responsible for gout. The uric acid will form crystals in the joints, which causes a type of arthritis called gout. So every animal has nitrogenous waste that they have to get rid of. Your amino acids that build up proteins and your nitrogenous bases that are part of the nucleotides that build nucleic acids, which are DNA and RNA, those all contain nitrogen, and so anytime that you're degrading them, you end up with nitrogenous waste, which would be these amine groups that you have to get rid of. If you're a fish, you just convert that amine group to ammonia and it just floats away in the water. It's not really a problem at all for you. If you live on the land, then you have to do something a little bit more special. So mammals will be converting, and other animals, mammals will be converting the amine groups to urea and excreting it in the urine. If you're a bird, on the other hand, you're going to be converting them mostly to uric acid. So the uric acid has a white appearance. It's not very water soluble, so they can't really pee that out. And so what they do instead is defecate it out. So if you've seen the bird poop and it has like the white stuff, that white stuff is the uric acid that they are getting rid of. You, being a mammal, don't like to do that. You urinate it out instead as urea. You do make some uric acid still. You're able to urinate out some uric acid, just not a lot. So that's why you're mostly focused on converting your nitrogenous waste into urea. It's easier to eliminate. And this picture here is just some uric acid crystals that were actually eliminated in urine. Another waste product that's commonly found in urine is creatinine. That's from, oh, this is actually a typo. It's from, I'm going to change that. So you probably won't even see it as a typo. It's going to be fixed. That's creatine phosphate that comes from, creatinine comes from creatine phosphate. Creatine phosphate is used as a source of energy in your skeletal muscles. They can transfer that phosphate group on the creatine to ADP, which generates ATP. And then you have to do something with that creatine, and in general you will be converting it to creatinine and eliminating it in the urine. And then in addition to the nitrogenous waste and the creatinine, you're going to also have any other little toxins that were in your blood that are soluble in water coming out in the urine. In order for some waste product to be eliminated by the urine, it just has to be water soluble. So a lot of the time when your liver is detoxifying, well, toxins or drugs that you've eaten, like medicinal drugs, pharmaceuticals, its goal is to convert it not to something that's not toxic necessarily, but to something that is more water soluble. And the point of that is so that it can come out in the urine. So the liver and the kidneys are generally working together to detoxify and eliminate any toxin that you consume. So filtration by the renal corpuscle is measured using the glomerular filtration rate, or the GFR. The GFR, or the glomerular filtration rate, is how much filtrate is made every minute. And it's about 125 milliliters of filtrate formed per minute if you're looking at both of the kidneys together. So if you're looking at just one kidney, it would be half of that value. So that's very large. If you remember that you only have like 4 to 5 liters of blood in your body, depending on how big you are, then you're going to be filtering all of that many times in a day. Actually, roughly 60 times per day, all of the plasma in your body would be filtered through the kidneys, through the renal corpuscle. So one implication of that, of course, is that you need to be returning most of that back to the blood. You can't be losing all of the plasma in your blood 60 times per day. Of course, that's insane. So most of that is filtered out needs to go back. It needs to be reabsorbed in tubular reabsorption. But of course, the waste products will not be reabsorbed. Only the things that you actually need, like the water and some of the electrolytes and such. So it's a very high rate. One of the reasons why it's so high is because you have a lot of surface area in the filtration membrane. So you have very, very many little renal corpuscles. The cortex is full of renal corpuscles, and the tubules of the nephrons, of course. And each corpuscle has multiple loops of the glomerulus because that's a little capillary bed. So you have a lot of surface area in there for filtration. And that filtration membrane also has a high permeability, which means things pass through it very easily. Anything that's small will very easily pass through it. The glomerular filtration is driven by a pressure gradient between the blood and the capsule lumen, the lumen of the glomerular capsule. And there's a very similar process to what drives water to enter or exit the capillaries in most tissues in the capillary bed serving any given tissue. So as you remember, anytime you have blood passing through a capillary, you're going to be losing some of the water from that blood. You have the capillary hydrostatic pressure driving water out. That's basically just the blood pressure in the capillary that pushes water out. You also have the tissue hydrostatic pressure. That would be pressure generated by fluid that's already in the tissue, pushing water back in. But in most of your tissues, that pressure is extremely low. Well, basically zero. Then you also have osmotic pressures. So you have the capillary osmotic pressure that is pulling water in. So all of the solutes that you have concentrated in your blood are pulling water or attracting water in to them by osmosis. Then you also have the tissue colloidal pressure, which would also be called the interstitial colloidal pressure, well, the interstitial osmotic pressure and the interstitial hydrostatic pressure. So the interstitial osmotic pressure, or the tissue colloidal pressure, is just all the solutes that are in the tissue itself, in the interstitial space, the little spaces between the cells, that are pulling water towards them by osmosis. So you have overall water leaving the capillaries on the arterial end, and then you have some water going back on the venous end because the blood pressure in the capillary has dropped by the time you get to the venous end by a lot. Overall, you have some water leaving the capillaries, and that extra water would get sucked up or pushed out actually into the lymph nodes. Well, into the lymphatic vessels, and then travel through the lymph nodes and ultimately be returned to the blood. So you have the same basic forces applying in the kidneys, just with the balance of them being a little bit different. Whoops. So all of those four forces that are pulling water in or out, or pushing water in or out of the capillaries, can be summarized using the net filtration pressure, or the NFP. So for most of your tissues, in all cases, the net filtration pressure equals the blood hydrostatic pressure. That's your blood pressure, which would be actually the blood pressure in the capillaries themselves, plus the interstitial osmotic pressure. That is the pressure shown here in green. That is all these solutes that you have in the tissues pulling water towards the tissues by osmosis. Then you have minus the blood osmotic pressure, which would be the solutes in the blood, primarily albumin, pulling waters into the capillaries, and minus the interstitial hydrostatic pressure, which is the pressure of the fluid in the tissue. So that's a little complicated, but we can simplify that. So first, we talk about the oncotic pressure, or the OP. The oncotic pressure is also called the colloid osmotic pressure, or the COP. So the book frequently calls it the COP, or the colloid osmotic pressure, but I like to use oncotic pressure because it is fewer words. The oncotic pressure is the blood osmotic pressure minus the interstitial osmotic pressure. So it's summarizing all the osmotic forces that are attracting water to the capillary or to the tissues. So you have solutes in the blood attracting water into the blood by osmosis. You have solutes in the tissues attracting water to the tissues by osmosis. If you combine those together, you get the oncotic pressure. In general, the blood has more solutes than the tissues do. So the blood osmotic pressure is going to dominate, and that's why it's the blood osmotic pressure minus the interstitial osmotic pressure. So for most of your tissues, you can simplify the NFP to the blood pressure minus the oncotic pressure. That's because the tissue hydrostatic pressure for most of your tissues should be 0 or very, very close to 0. Anytime that pressure rises, all the extra fluid would go into the lymphatic vessels and the pressure would drop back down again. So the blood pressure is, a non-zero value. The tissue hydrostatic pressure is almost 0, so it cancels out. And then you're combining both of the osmotic pressures into one thing, the oncotic pressure. For the kidneys, it's a little bit different. So in the kidneys, the net filtration pressure is simplifying to the glomerular hydrostatic pressure, which is the blood pressure in the glomerular capillaries. So in these capillaries here, the blood pressure right in there, that's the GHP, or the glomerular hydrostatic pressure. So you have the GHP minus the GOP, which is the glomerular oncotic pressure. So that would be taking into account the osmotic pressure within the capillaries, attracting water to the capillaries, and the osmotic pressure in the glomerular capsule, which would be coming from the filtrate, attracting water into the filtrate. You combine those two things together to get the GOP, or the glomerular oncotic pressure. And then, so you have the GHP minus the GOP, and then minus the CHP, which is the capsular hydrostatic pressure, or the pressure of the filtrate in this glomerular capsule. So it's the same basic set of forces, but the location is different. So instead of having just tissue here, you have an empty space, the glomerular capsule, that is filled with filtrate, and so the pressures end up being slightly different in their values. Right, so the pressure of the filtrate inside of this capsule is not zero. So you have the GHP, the glomerular hydrostatic pressure, pushing water out into the filtrate. Then you have the GOP, the glomerular oncotic pressure. That's composed of the glomerular, or the blood osmotic pressure pulling water into the capillaries, and the filtrate osmotic pressure pulling water out of the capillaries. But in this case, actually, you have an extra high contribution from the osmotic pressure in the blood itself, from that albumin that's in there, because you have so much water leaving the capillaries and entering the filtrate. So the blood in these capillaries, as the water leaves, is becoming very concentrated. You have less water, and you still have a lot of solutes because of all the proteins that stay behind, in particular the albumin. The albumin can't cross into the filtrate, so that's providing a lot of osmotic pressure. So you have that GOP attracting water back into the glomerular capillary. And the book again calls it the GCOP instead, the glomerular colloid osmotic pressure, but I like GOP because, again, it is shorter. Then finally, the capsular hydrostatic pressure, or the pressure of the filtrate inside of this capsule, is higher than normal. It's not, it's not zero, because you actually have a significant amount of fluid in here. It's also going to be moving out into the tubules, of course, and ultimately to the collecting ducts and then into the ureter, but there's only so fast that can happen. It's being filtered out very quickly. So if we zoom in, you can see those forces kind of summarized here. So you have the GHP, this would be the capillary side, and here you have the capsule side. So this is the GHP, the blood pressure inside of the glomerular capillary, that's pushing fluid out through the filtration membrane. Then you have the GOP, or the GCOP, that is primarily dominated by the osmotic pressure inside of the blood, which is becoming hyper-concentrated because it's losing water. So that is attracting water back into the blood by osmosis. And then you have the CHP, or the capsular hydrostatic pressure, that is also pushing water back into the blood. So that's the pressure of this fluid of the filtrate pushing the water back into the blood. However, if you add it up, the CHP plus the GOP, that equals about 40 millimeters of mercury of pressure, whereas the GHP is about 50. So the GHP is going to win every time, and you're ultimately going to have water leaving the blood and entering the filtrate, and of course solutes coming with it. So the NFP, or the nut filtration pressure in the kidneys, is about 10 millimeters of mercury, although of course your body is able to vary that to control how quickly you're forming urine. And now we'll talk about how exactly the body does vary that to control how quickly you're forming urine. So the glomerular filtration rate is that's the start of the formation of your urine. That's where the filtrate is initially forming from the blood. So if you're going to control how much urine you're making, you want to control it at the source, which is the GFR. So you have to control the GFR to gain control of how much urine you are producing. You have three things that are regulating the GFR. You have the nervous system, you have the endocrine system, and then you have autoregulation by the local tissue itself. each one of those systems or ways of regulating the GFR is relying on the same mechanism. In each case, the mechanism for changing the GFR is based on vasoconstricting or vasodilating the afferent and efferent arterioles. So remember, the afferent is going to, that's the arteriole leading into the glomerulus capillary bed, and then the efferent arteriole is going from the glomerulus capillary bed, so it's leading away from it. So if you constrict the efferent arteriole, then blood is going in to the glomerulus at the same rate, but it's hard for it to go out. There's more resistance, there's more vascular resistance when it's trying to get out. So it can't get out quite as fast. That means it backs up in the glomerulus a bit, and that increases the GHP, and if you increase the GHP, you're going to increase the GFR. So that means that the blood pressure inside of this capillary bed is going up because you have more blood pressure that pushes the water out, and you end up forming more filtrate more quickly. On the other hand, if you were to vasodilate the efferent arteriole, the opposite thing would happen. You have blood coming into the glomerulus at the same rate, but it's very easy now for it to drain out of the glomerulus. So instead of getting filtered out into the glomerular capsule, a lot of that fluid is just leaving in the efferent arteriole now. There's more space for it there. So that means the GHP will go down, the blood pressure inside these capillaries, which is the GHP, will go down, and anytime the GHP goes down, the GFR goes down as well, and you'll be forming less urine. If you're affecting the afferent arteriole instead, you have the opposite effects. So if you vasoconstrict the afferent arteriole, that means that now it's harder for blood to get into the glomerulus. There's more resistance that this blood is experiencing as it's trying to move into the glomerulus. So it doesn't come in quite as fast, but it's still leaving just as easily as ever. So that means that the GHP is going to go down. The blood pressure inside of these capillaries is going to go down. Anytime the GHP goes down, the GFR goes down. On the other hand, if you were to dilate the afferent arteriole instead, now it's very easy for blood to enter the glomerulus. It flows in really easy. You get a lot of blood entering the glomerulus. That means that the blood pressure is going to go up in there because it's still just as hard for it to leave as ever. You've made it easier to enter, but you have not made it easier to leave. So it's going to get backed up in here a bit, and that's going to increase the blood pressure in the glomerulus. Anytime that goes up, that means that the GHP is going up because the GHP is the blood pressure inside of the glomerulus. And if the GHP goes up, the GFR goes up as well. So for the efferent arteriole, constricting it means the GHP goes up and the GFR goes up. For the afferent arteriole, dilating it means the GHP goes up and the GFR goes up. For the efferent arteriole, dilating it means the GHP goes down and the GFR goes down. For the afferent arteriole, constricting it means that the GHP goes down and the GFR goes down. They have opposite effects on the GHP. So anytime you're trying to control the GFR, you're going to be doing one of those things: vasoconstricting the efferent or afferent arterioles, or vasodilating the efferent or afferent arterioles. By doing that, you can alter the GHP and therefore alter the GFR. So first we'll talk about autoregulation, that is local regulation of the GFR by the nephron and the surrounding kidney tissue itself, and by the arterioles themselves. So the main purpose of the autoregulation is just to provide a stable GFR, so that even if your systemic blood pressure is changing because you had stood up or something, or just because you're going through your day and blood pressure is just going to change a bit as you're going through your day, the GHP should not change, even though the blood pressure is changing, and it's autoregulation that accomplishes that. So the autoregulation mechanisms, you have two of them: the myogenic mechanism and the tubuloglomerular feedback mechanism. Those work together, and they're able to maintain a stable GFR in a range of blood pressure, of systolic blood pressure between 80 millimeters of mercury and 180 millimeters of mercury. So your normal blood pressure is well within that range, and your blood pressure can increase quite a bit and you can still maintain a steady GFR. If your blood pressure does drop below 80 millimeters of mercury, then the autoregulatory mechanisms begin to fail, and your GFR is going to start dropping as well. If your systolic blood pressure drops below 70 millimeters of mercury, then there's not going to be enough GHP left over to drive glomerular filtration, and the GFR is going to crash to basically nothing, and at that point you will be in kidney failure when they're not making any urine or filtering the blood. So first we'll talk about the myogenic mechanism. That is very similar to myogenic mechanism that is used to maintain tissue perfusion or in local capillary beds in basically any tissue in the body. So it's all about the muscles in the walls of your arterioles contracting based on how much stress you have in the arteriole. So if the blood pressure goes up, that means that the arterial wall is going to stretch some more to accommodate the extra pressure of the blood. Or it's just the blood will be exerting more force on the walls of the arteriole, and that extra force makes the wall stretch. So you're going to have stretch receptors in the walls that sense that they are stretched, and the response will be that the smooth muscle is going to constrict. So the smooth muscle will contract in the walls of the arteriole, and that causes vasoconstriction. So if you have an increase in blood pressure, that gives you stretch, that gives you muscle contraction, that gives you vasoconstriction. That's the myogenic mechanism. And it works the opposite way as well. If your blood pressure goes down, then the arterial walls will not stretch as much, and the stretch receptors will sense that you have less stretch in the walls, and they're going to relax the smooth muscles, and that causes vasodilation. So you can see in this graph, it's kind of showing how that works. On the top graph, we have the arterial pressure, which is what they were manipulating in this experiment. Then here you have the arteriolar radius, which is just how big the arterioles are. That's measuring the degree of vasoconstriction or vasodilation in the arterioles. And then here you have the capillary pressure. So that would be the pressure in the capillary bed that is coming after the arterioles. So if the arterial pressure jumps up, you're going to have that increase in pressure cause a stretch in the walls of the arteriole, so that's going to get bigger momentarily. Then those stretch receptors will activate, and then the smooth muscle will contract, and that causes the arteriolar radius, or the diameter of the arteriole, to drop. So your vessels or your arterioles are going to constrict. Then downstream from that in the capillaries, you can see that the capillary pressure does rise a bit because of this rise in arterial pressure. But then once the arterioles start constricting, then you have more resistance, you have less blood entering the capillaries, so the pressure in the capillaries is going to go back down to normal. Even though the pressure in the arterioles has not gone back down to normal, it's still high. So this mechanism would be operating extensively in the afferent arteriole. If your blood pressure goes up, then the pressure in that afferent arteriole will go up. It's going to be stretched. The smooth muscle in its walls will constrict. So now you have a vasoconstricted afferent arteriole. Anytime that happens, you're going to have less blood entering the capillaries, and so that maintains a steady capillary pressure. You would have more blood entering because of the increase in arterial blood pressure, but instead that's been counteracted by the vasoconstriction, so that evens out to regular, and that you still will have a regular GFR at that point. The next mechanism of autoregulation is tubuloglomerular feedback, which is based on the action of the juxtaglomerular apparatus, or the JGA. So we had mentioned the juxtaglomerular apparatus before when we talked about microscopic kidney anatomy, and now we're going to talk about what it does. So as you remember, that would be found right here. This is the renal corpuscle. You have the proximal tubule leading away from it. That's going to wind around, go down into the nephron loop, come back out, and then start winding around again as the distal tubule. And you have this one loop of the distal tubule that comes close to the renal corpuscle and to the glomerulus, which is why it's called the juxtaglomerular apparatus. You have two cell types within the juxtaglomerular apparatus. You have the macula densa cells, which are shown in green here in this diagram. So those are part of the simple cuboidal epithelium that is lining the distal tubule. Then you also have the juxtaglomerular cells, or the JG cells, which are shown here in blue and orange. Some of them, the ones that are in blue, are actually specialized smooth muscle cells that are lining the walls of the afferent and efferent arterioles. So the way that this works is that anytime the GFR goes up, that means that you're going to be forming more filtrate. That means more filtrate will be entering the tubules. More filtrate will be coming through the distal tubule right here. And the distal tubule, one of its jobs is to reabsorb electrolytes from the filtrate. So it would be reabsorbing sodium and chloride ions and returning them ultimately to the blood. And so all of these cells that are lining the distal tubule are doing that. When the macula densa cells do that, they will release a signaling molecule. So if you have more filtrate formed because the GFR went up, more filtrate coming through the tubules, then the macula densa cells will be reabsorbing more sodium and chloride than they were before. When they absorb more sodium and chloride, they release more ADO. ADO stands for adenosine nucleoside, and it's like something that's formed from ATP. So actually these cells are releasing ATP and then it's being converted into ADO, or adenosine nucleoside. And that acts as a paracrine signaling molecule, which means that it diffuses through the local tissue and does not enter the blood, but it does carry a signal or a message. So in this case, that ADO is going to be diffusing to these cells, to these juxtaglomerular cells, and binding to receptors on them. Ultimately, that causes the smooth muscle ones to constrict, especially in the afferent arteriole. So that causes vasoconstriction of the afferent arteriole if the GFR had gone up. If the afferent arteriole constricts, you'll have less blood entering the glomerulus and the GFR will drop. So that's the basic way that the tubuloglomerular feedback mechanism works. This is kind of more detailed diagram showing that. And you very much don't need to know the details in this diagram. But here you have the macula densa cells that are lining the distal tubule. Here you would have the lumen of the distal tubule. Then this is the interstitial space, and here you have the juxtaglomerular cells. There's actually 3 different types of cells here. That's one, that's another, and then on the top you have another. Those are all different types of juxtaglomerular cell. And the ones that are actually down here, those are the smooth muscle cells. Those would correspond to the ones in blue. The top layer would correspond to the cells in orange up here. And then right here you would have the endothelium of the afferent arteriole. So when you have sodium and chloride being taken into these macula densa cells, they will start releasing ATP, and then that gets converted into ADO, or adenosine nucleoside, and that binds to its receptor on these juxtaglomerular cells, and that creates a cascade of events that ends with these smooth muscle juxtaglomerular cells constricting, which constricts the afferent arteriole and reduces blood flow into the glomerulus, which lowers the GHP and therefore the GFR. Actually, this would always be happening, even kind of, at rest or under normal circumstances all the time. So you'll always have those macula densa cells reabsorbing some amount of sodium chloride and releasing some amount of ATP, which is converted to ADO and causes some amount of vasoconstriction in the afferent arteriole. If the GFR goes up, the sodium and chloride go up, ADO goes up, vasoconstriction goes up. If the GFR goes down, then the opposite thing happens. Then the macula densa cells are reabsorbing less sodium and chloride, and they're going to release less ATP, which is not converted to ADO as much since there's less of it. And then you're going to have less ADO signaling and less vasoconstriction. So those specialized smooth muscle cells, those specialized juxtaglomerular smooth muscle cells will actually relax instead of constricting. And then you're going to have vasodilation of the afferent arteriole, which then causes more blood to enter the glomerulus, which increases the GHP, which increases the GFR. So in this way, you maintain a steady GFR even in the face of changing blood pressure. So next we'll talk about how the endocrine system is regulating the GFR. You have two main hormone systems that regulate the GFR. First you have the RAAS, the renin-angiotensin-aldosterone system, and next you have the hormones from the heart, ANP and BNP. So for the renin-angiotensin-aldosterone system, that one is, or the RAAS, that one is of course the most complicated, so we'll talk about that in some detail. The ultimate effects of that system are to raise the blood pressure. The system has multiple ways of doing that. It's going to increase the vascular resistance, and it's going to increase the blood volume. In addition to kind of controlling the blood pressure, the system is used to maintain water and electrolyte homeostasis. In terms of its effects on the GFR, it would raise the GFR. The ANP and BNP system is a little different. ANP is atrial natriuretic peptide, BNP is brain natriuretic peptide. Even though it's called brain natriuretic peptide, it still comes from the heart. They didn't know where it came from when they named it, so they named it wrong. Both of those hormones have very similar effects. Generally, they would be lowering the blood pressure by lowering vascular resistance and lowering blood volume. But in terms of the GFR, they would actually be raising the GFR. So both of these hormone systems act to increase the GFR, even though they have opposite effects on blood pressure. And we'll get into the details of how that occurs. So first, the renin-angiotensin-aldosterone system, or the RAAS. Its general job is to maintain the blood pressure and your water, ion, and pH homeostasis. It all starts in the kidneys with the hormone renin. So the kidneys will release renin if blood pressure goes down or if the sympathetic nervous system stimulates them to. It's actually released by the juxtaglomerular cells themselves. It also responds to the tubuloglomerular feedback. So if the ADO goes down, if the GFR goes down, then the ADO will also go down. So if the GFR or the ADO go down, that's also going to stimulate those JG cells to make renin. So they make it in response to low blood pressure, to low GFR, to low ADO, or to signaling from the sympathetic nervous system. The renin hormone is considered the rate-limiting hormone for the system, which means that how much renin you have is determining how active the system is. You have multiple hormones in this system, but the activation of all of them, or the release of all of them, is dependent on how much renin you have. So renin's job is actually just to convert angiotensinogen to angiotensin 1. Angiotensinogen is made in the liver and released into the blood. It is inactive until it's inactive, and then renin converts it to angiotensin 1. Angiotensin 1 is actually still inactive, so it's a little bit confusing. Angiotensin 1 still has to be converted more to be activated. That actually happens in the lungs. Angiotensin 1 would be circulating in the blood. When it passes through the lungs, you have this enzyme bound to the endothelium of blood vessels in the lungs, which is called ACE. That is angiotensin converting enzyme, and its job is to convert angiotensin 1 to angiotensin 2. Angiotensin 2 is actually active, and it is a proper hormone that has, effects. ACE itself is not considered a hormone. Renin is considered a hormone. Its only real job is to help activate angiotensin 2. ACE also helps activate angiotensin 2, but it is not considered a hormone, and the reason is that it does not travel in the blood. It's actually attached to the endothelium, so it's not free in the blood, so it's not a hormone. Then angiotensin 2 has some effects of its own on different body tissues. One of the effects that it has is to stimulate the release of aldosterone from the adrenal cortex, which, as you remember, the adrenal glands are sitting right on top of the kidneys. So let me go through some of these diagrams I have. Here you have another view of the JGA, of the juxtaglomerular apparatus. This would be the renal corpuscle here, with the proximal tubule leading away from it. This is the glomerulus. So here you have your afferent arteriole. This is the efferent arteriole. This is the curve of the distal tubule as it is coming close to the renal corpuscle. So right here you would have your macula densa cells that are part of the juxtaglomerular apparatus. And then here you have your juxtaglomerular cells, or your JG cells. Some of them are the smooth muscle cells, where they're actually lining the walls of the arterioles, and some of them are not smooth muscle cells. So these JG cells would be responsible for making renin. And they will do it if your blood pressure drops. If your blood pressure drops, then your GFR will probably drop as well until the autoregulation kicks in. If the GFR drops, they will make it. If ADO is released less by these macula densa cells, they will make renin, and ADO would be released less if you had less GFR. And then finally, here you have axons of postganglionic neurons in the sympathetic nervous system that are coming in to contact some of these JG cells. And if these nerves signal these cells, then they will start making renin. So you have the liver producing angiotensinogen. The liver is actually always producing some amount of angiotensinogen, so you always have angiotensinogen in your blood, but it's not active. It doesn't do anything until renin activates it. So if you have that drop in blood pressure, drop in ADO, drop in GFR, or SNS stimulation, then renin will be released by the kidneys. It's going to activate angiotensinogen to angiotensin 1. Angiotensin 1 is still inactive actually, but it travels in the blood. It goes through the lungs, and it is activated fully by angiotensin converting enzyme, or ACE. ACE will activate angiotensin 1 to angiotensin 2, which is actually an active hormone that has effects. ACE is actually always in the lungs. You just have like a set amount of ACE attached to the endothelium in the pulmonary vessels of the lungs. And you always have angiotensinogen being released by the liver. So you always have all these components except for renin, and that's why renin is rate limiting. The RAAS will activate if renin is released. If renin is not released, it will not activate, because of course you need angiotensin 1 in order to have ACE converted to angiotensin 2, but you cannot get angiotensin 1 if you do not have renin. Instead, you would just be stuck with angiotensinogen. So as soon as renin is released, the existing angiotensinogen is converted to angiotensin 1, the existing ACE converts that to angiotensin 2, and now your RAAS is active. This angiotensin 2 has different effects. It affects the brain by stimulating thirst. It affects the blood vessels by causing vasoconstriction, which increases vascular resistance. And it affects the adrenal gland by causing the adrenal cortex to release aldosterone. And the aldosterone and angiotensin 2 both have effects on the kidneys, which we're going to talk about right now. Ultimately, all of these effects are elevating blood pressure. So when you stimulate thirst in the hypothalamus, then you start drinking. That raises blood volume and therefore blood pressure. When angiotensin 2 causes vasoconstriction, that raises vascular resistance, which raises blood pressure. And actually, angiotensin 2 and aldosterone both cause water retention and sodium retention in the kidneys, and that raises blood pressure as well by raising blood volume. Some more details on angiotensin 2. Angiotensin 2 can be abbreviated as Ang2. One of the things it does, again, is cause aldosterone to be released from the adrenal cortex. It also has non-tropic effects. That would be effects beyond causing the release of a hormone. So it causes vasoconstriction. It actually constricts all of the systemic blood vessels, which raises vascular resistance and therefore blood pressure, but it especially constricts the efferent arterioles. Remember, when the efferent arterioles are constricted, that is going to raise the GHP because blood cannot get out of the glomerulus as well. So it's constricting everything, but it's constricting the efferent arterioles more. So you have an increase in the GHP as a result of that vasoconstriction. The increase in GHP is going to increase GFR. Another thing angiotensin does is increase the retention of water and sodium and chloride in the kidneys. So it actually would be affecting the ions themselves. It would be directly affecting retention of or reabsorption of the sodium and the chloride, and then water will just follow the sodium by osmosis. So in general, in the kidneys, when you're talking about reabsorption in the tubules, wherever the sodium goes, water is going to end up following it if water is able to follow it by osmosis. So that means that you are reabsorbing more water and putting less of it into your urine. Ultimately, that has the effect of at least preserving your blood volume. But as you continue to drink, of course, then you're going to be increasing your blood volume. So in general, anytime you have water retention in the kidneys, blood volume will go up because you're continuing to drink water still, but you're not making as much urine. And if blood volume goes up, then blood pressure will go up. And also, angiotensin II acts directly on the brain, on the hypothalamus, which is the part of the brain that's primarily responsible for regulating thirst and fluid intake, and it stimulates thirst, which makes you go drink some water. And when you drink water, that increases your blood volume and therefore your blood pressure. So overall, all of these effects are increasing the blood pressure. The vasoconstriction is especially increasing the GHP, but also anytime that blood pressure goes up, GHP will go up, and then you're going to have an increase in the GFR. Next, we'll talk about aldosterone. The effects of aldosterone, the main effects of aldosterone are on the kidneys. Aldosterone is causing more ion channels and ion pumps to be inserted into the membranes of the epithelial cells lining the kidney tubules. And when that happens, you're going to have more ions moving one way or another, either being reabsorbed by the tubules or secreted by the tubules, depending on the ion. And we'll go into the details of which ions are reabsorbed or secreted where, in a little bit. Overall, aldosterone is going to be increasing the reabsorption of sodium and chloride, so increasing retention for sodium and chloride. When that happens, when you're reabsorbing sodium, as I had mentioned, water will always be following it by osmosis. So if you retain sodium, you're going to retain water too. When it comes to potassium, you actually have the opposite effect. Aldosterone would be increasing the excretion of potassium. So it decreases reabsorption of potassium, so more potassium that's in the filtrate ends up in the final urine product. You can remember what effects it has on what ions by remembering where each ion is supposed to be most concentrated. So sodium and chloride are supposed to have a high concentration outside of the cells and a low concentration inside of the cells, as you may remember from ANP1. So in order to achieve that, aldosterone is important, or aldosterone that helps you achieve that by retaining the sodium and the chloride, so you can have more of it in the interstitial fluid outside of the cells. Potassium, on the other hand, needs to be high inside of the cells and low outside of the cells. So when aldosterone causes potassium to be reabsorbed less and excreted more, that means that the concentration of potassium in the interstitial fluid is dropping. So you end up with less outside of the cells, which is exactly what you wanted. So aldosterone is helping you maintain those ion concentrations at where they need to be. It also increases the excretion of hydrogen ions. Hydrogen ions are protons. Anytime that you have more hydrogen ions, you would have a lower pH or a more acidic solution. If you are excreting hydrogen ions, then you are raising the pH and having a less acidic or more basic solution. The pH of the blood ideally should be 7.4. 7 is neutral and 7.4 is just slightly basic, which you could also say is slightly alkaline. In order to get to 7.4 from 7, 7 is the natural pH of water. In order to get up to 7.4, you have to get rid of some protons. You have to get rid of some hydrogen ions. And so aldosterone is helping you do that by excreting those things in the urine. So you can remember its effects on hydrogen ions as well by remembering that the pH of the blood is slightly basic, and to get there you would need to get rid of some hydrogen. And then finally, you can remember its effects on water because water always follows sodium. So if you just remember that where the sodium goes, the water will go, then you will remember that water is retained when aldosterone is secreted. So those are the effects of aldosterone on the kidneys: to increase retention of sodium, chloride, and water, and to increase excretion of hydrogen and potassium ions. That water retention is going to end up increasing your blood volume because, of course, you continue to drink even though you're not making as much urine. When your blood volume goes up, the BP goes up. When the BP goes up, the GHP goes up. And when the GHP goes up, the GFR goes up. And that is the way that aldosterone increases the GFR. So here's a summary of the RAAS, and due to our lovely current events, I'll also mention how it's connected to COVID-19. So here you have the summary of the RAAS. Here you can see the systemic blood pressure is going down. That causes GFR to go down. That causes the JG cells to release renin. That would also cause the ADO to go down, and then those cells release renin. Also, the sympathetic nervous system could signal the JG cells to release renin. The liver, meanwhile, has already released angiotensinogen, always releasing angiotensinogen. So you have angiotensinogen in the blood all the time from the liver. Then when you release renin in response to the low blood pressure, low GFR, low ADO, or SNS signaling, then you're going to have that angiotensinogen converted to angiotensin 1. Angiotensin 1 travels in the blood vessels to the lungs, where you have ACE. ACE is lining the walls of those blood vessels. It is not in the blood, it's not free in the blood, so it's not a hormone. And its job is to convert angiotensin 1 to angiotensin 2. Angiotensin 2 is active. And angiotensin 2 will then go and, it has all of its effects. It's going to cause you to become thirsty by affecting the hypothalamus. It's going to cause aldosterone to be released from the adrenal gland. Then aldosterone will be affecting ion homeostasis, increasing retention of sodium and chloride, and therefore water as well, increasing excretion of hydrogen ions and potassium. The angiotensin 2 will also itself be increasing reabsorption of sodium and chloride, and then the water will be following the sodium, so you're going to increase water retention as well. And finally, angiotensin 2 will be constricting all of your blood vessels, raising vascular resistance. So that increase in vascular resistance increases blood pressure. The increase in water retention and the increase in thirst, all of those things act together to raise the blood volume, and all of those things raise blood pressure. So now we've countered the initial decrease in the blood pressure. Another thing that angiotensin 2 would be doing is extra vasoconstriction in the efferent arteriole. So the afferent arteriole will constrict just like every other blood vessel in the body that has smooth muscle, but the efferent arteriole will be constricting more. So you're going to be raising the GHP even more than you're raising the regular blood pressure, and that's going to mean that GFR goes back up. And then finally, I'll just mention again that renin is the rate limiting step here in the whole RAAS system, because you always have angiotensinogen released, and you always have ACE lining the vessels in your lungs. So you're always ready to kind of make angiotensin 2 in terms of having the ACE that you need and having the starting material, the angiotensinogen. The only thing you don't have at rest is the renin itself. So if you make renin, then you're going to convert angiotensinogen to angiotensin 1, and then angiotensin 1 will be converted to angiotensin 2. If you don't make renin, that will not happen. Okay, so that's the RAAS. Now for COVID-19. So here you have, well, this is actually, I guess this is angiotensin 1, and then showing it being activated by ACE to angiotensin 2. Then angiotensin 2 has receptors, and it has receptors for it actually in many places in the body. It has a lot of effects. It has the ones that I talked about, but it also has some other effects too. All of these hormones have very many effects, and we're really only focusing on the primary ones. But if you want to get into the details of the hormone, you would discover many other functions as well. And so some of those functions that angiotensin II has in the lungs are to cause the vasoconstriction that we had already talked about, but it also causes other things. It increases the vascular permeability for one thing. And if it binds to this receptor in the heart, that can also cause what they've referred to as adverse myocardial remodeling. That means reshaping of the muscular wall of the heart in a way that is not good for you. And it also seems to have some association with acute lung injury. It's not really clear how that works, but anytime that you have infection in the lungs and you're making angiotensin II, that makes it worse. The damage to the lungs will be worse if you have a lung infection and you have angiotensin II. It's not really clear why that is, but you know, it's something. It sure is something. You also have another enzyme in the lungs called ACE2. So actually ACE1 is the regular enzyme that we're used to that activates angiotensin 1 to angiotensin 2. At the same time, you have this other form of that enzyme called ACE2, which does a different thing. Instead of activating angiotensin, it deactivates it. So it takes angiotensin 1 and degrades it to this angiotensin 1,9 product. It takes angiotensin 2 and degrades it to this angiotensin 1,7 product. These things are not active and they cannot be activated. They're just going to be degraded and gotten rid of. So it's thought that under normal conditions, ACE2 is acting to kind of limit the amount of angiotensin 2 signaling that you have through this angiotensin 2 receptor. So that way you don't have too much angiotensin 2 signaling, you don't have too much vasoconstriction or vascular permeability, you don't have too much of that adverse cardiac remodeling. And so ACE2 is actually protective. It helps you balance the amount of angiotensin 2 that you have active at any given time. On the other hand, ACE2 is actually the receptor that is being used by the coronavirus to enter your cells. So SARS-CoV-2 is like the fancy pants science name for the novel coronavirus. SARS-CoV-1 is actually the SARS virus that was spreading in the early 2000s and was a lot more deadly than coronavirus, but also was not nearly as transmissible, so it didn't cause a pandemic. But it was still very bad in the areas where they had an epidemic of SARS. Both of those viruses are using the ACE2 receptor to enter cells. So they will bind to ACE2 and then they can get inside of the cell and they're infecting that cell now. If you have a cell that does not have the ACE2, then they're not able to get inside and those cells would be safe. But those cells would not be safe from angiotensin II. So it's a little strange. There's been some suggestions that people who are on ACE inhibitors or just different drugs that inhibit the RAAS system should maybe stop taking those drugs because of this connection between ACE and the coronavirus. It's not really clear if that's actually the case. Of course, if you're taking that type of drug, there's a reason why you're taking it. And if you stop taking it, you're going to have probably some withdrawal effects from stopping taking the drug that you had been eating every day for a long time, as well as whatever disease you were taking. You were using the drug to control, that disease would not be controlled anymore. Those drugs are used a lot to control hypertension. So there has been some suggestion that you should stop taking those drugs, but there's also the possibility that you should not stop taking those drugs because they actually reduce the amount of angiotensin 2 that you have, and angiotensin 2 is associated with greater lung damage and actually heart damage in the case of infection. So it may actually be protective, it may not be protective. You know, we don't really know yet. We really just don't have enough information yet to know for sure. But bottom line is, don't stop taking whatever drugs you're taking. Don't do that. Just be careful. Okay, so now for the next hormone system that regulates the GFR, ANP and BNP. Atrial natriuretic peptide, or ANP, is released from the atria of the heart in response to high blood pressure. Anytime the blood pressure goes up, that's causing extra stretch in your blood vessels. It also causes stretch in the heart chambers. So the atria would stretch in response to that high blood pressure. Then you have stretch receptors in the walls of the atria that activate and cause the atria to start making and releasing ANP. Then brain natriuretic peptide is very similar, but it's coming from the ventricles. So it's definitely misnamed. It should probably be called ventricular natriuretic peptide instead. But you know, I guess the name they gave it when they didn't understand where it came from just kind of stuck, and that's what everybody thinks of it as now. Its effects are basically the same as ANP, except that it's coming from the ventricles instead of the atria. So it's also released in response to high blood pressure and ventricular stretch. The effects of ANP and BNP are to, well, I mean, in general, their effects are to cause vasodilation and to cause water excretion in the kidneys, so that you'll be making more urine, so your blood volume would go down. That lowers the blood pressure, and then with the vasodilation, you would have less vascular resistance, and that also lowers the blood pressure. So its overall effects on the blood pressure are to reduce it. It's released in response to high blood pressure and then lowers the blood pressure back down. But it has also special effects on the GFR. So you have, in response to ANP or BNP, vasodilation in the afferent arteriole, just like all the other blood vessels. But in the efferent arteriole, you don't have vasodilation. Instead, you have vasoconstriction. So with vasodilation of the afferent arteriole, that gives you more blood coming into the glomerulus, which would raise the GHP. And then with vasoconstriction of the efferent arteriole, that makes it harder for blood to leave the glomerulus. So that's also going to raise the GHP. And anytime the GHP goes up, then the GFR will go up. So it has an opposite effect on the blood pressure from the RAAS system, but the same effect on GFR to ultimately increase it. And in this diagram here, is showing that you're actually releasing ANP and BNP from the heart in response to stretch of the atria or the ventricles, also in response to other things. And then you would have those two hormones causing vasodilation in your blood vessels, which reduces vascular resistance and reduces the blood pressure. They're also going to act on the kidneys. They increase the GFR, and when they increase the GFR, they would actually be increasing water excretion. Because you're making more filtrate, you're going to be losing more urine and more water, therefore. And that reduction in the water, that increase in water excretion in the urine is going to reduce the blood volume, which then reduces the blood pressure. And you can also see that ANP and BNP are reducing the rate of renin release. So they actually have inhibitory effects on the RAAS. Actually, in general, the RAAS and the ANP-BNP system inhibit each other. So if you have one active, that's going to decrease activation in the other in general. And then finally, the nervous system is able to affect the GFR, again, by affecting the afferent and efferent arterioles. It's actually only the sympathetic nervous system that affects the GFR, not the parasympathetic. Overall effects of the sympathetic nervous system are to increase the blood pressure, and it does that in multiple ways. So it is raising the vascular resistance by causing vasoconstriction in many blood vessels serving the urinary, digestive, reproductive, and integumentary systems and most exocrine glands. It's also raising cardiac output. It does that in multiple ways. It increases the heart rate. It also increases stroke volume by increasing contractility. So all of those effects, contractility, heart rate, vasoconstriction, and all those different systems are ultimately raising the blood pressure. Another thing that the sympathetic nervous system does is trigger the release of renin from the juxtaglomerular cells. So those cells all have sympathetic nerves, sympathetic axons coming into them, and if those axons are stimulated with action potentials, the JG cells will release renin. And then you're going to have activation of the RAAS, which is also raising the blood pressure by raising vascular resistance and blood volume, and raising the GFR. The SNS has maybe interesting effects on the GFR. If you have a low level of activation of the sympathetic nervous system, in general, that will raise the GFR. You're going to get some renin being released, some activation of the RAAS, so some angiotensin II as well, and aldosterone. And you're going to get a lot of vasoconstriction in the efferent arteriole. Due to that angiotensin II. You would actually have vasoconstriction in all of those blood vessels serving the urinary system due to the sympathetic nervous system and to angiotensin II, but you're going to have extra vasoconstriction in the efferent arteriole. That means that overall the GFR will go up. On the other hand, if you have very high activation of the sympathetic nervous system, the type of activation that would occur if you had some form of emergency or were, doing a good exercise that day, then you're going to actually have the opposite effect on the GFR, and the SNS will decrease the GFR. In that case, the SNS will cause a lot of renin to be released and a lot of angiotensin II to be activated. And that means that you're going to have a lot of vasoconstriction, both due to the SNS itself and due to the angiotensin II. So that means that actually you're going to end up with so much vasoconstriction in the afferent arteriole that it doesn't really matter that the efferent arteriole is vasoconstricted anymore, and blood is going to have a very hard time getting into the glomerulus, and that is going to reduce the GHP and therefore the GFR. So low activation of the SNS gives you mostly vasoconstriction in the efferent arteriole, which raises the GHP and raises the GFR. High activation of the SNS is going to give you the opposite thing, more vasoconstriction in the afferent arteriole, or enough vasoconstriction in the afferent arteriole to reduce the GHP and therefore the GFR. So the image over here is kind of showing something that they've discovered. People who have hypertension that is not responsive to lifestyle changes and drug treatments like the ACE inhibitors, it seems that actually the sympathetic nervous system is playing a big role there, and specifically the sympathetic signaling to the kidneys. And why exactly that's occurring, it's not entirely clear, but in general, if you have a decrease in the oxygen supply to the kidneys, that might reflect a decrease in blood supply or some other problem. A decrease in oxygen supply to the kidneys is going to signal these viscerosensory nerves that are in the kidneys to tell the brain, that there's that oxygen problem. And then in response, the brain will send out sympathetic nervous system signals activating the sympathetic nervous system in the kidneys, and activating those JG cells to release renin. And then you're going to have actually all the activation of the sympathetic nervous system based on this signal from the kidneys to the brain. And in normal circumstances, that should be kind of balanced, so you don't have too much sympathetic nervous system activation, and when you don't need it anymore, it goes away. But in this resistant hypertension, it actually does not go away, and it's always active. So they're always having this vasoconstriction, this water retention in the kidneys due to the RAAS, and the increase in heart rate and contractility, which puts more strain on the heart. Just constantly. And one way that they've figured out that you can fix this is by preventing sympathetic nerves in the kidneys from sending their signals. So here you have the kidney. Well, this is a kidney. This is the ureter coming out of the kidney. And here you have the artery that's going into the kidney. So this would be the renal artery. And then in yellow here, you have sympathetic nerves that are going into the kidneys and are going to be contacting the JG cells. And if you can get rid of those nerves, then you can basically treat that hypertension without drugs. The surgery to cut those nerves is, of course, very invasive and dangerous. So it's nice if there's something else you can do. And they have found that there is something else you can do. You can insert this catheter into these arteries and wind it up into this renal artery, and just make sure that it's next to those sympathetic nerves. And then it has a little radio emitter on the end of it, which is right there. And you send these radio signals that impact the nerves and stop them from sending their action potentials. So you're actually deactivating the nerves without physically destroying them. And so it's a lot less invasive, a lot safer, and it can treat hypertension in some people who aren't responsive to drug treatments. So renal failure is centered around the GFR. Anytime the GFR drops by 50% or more, you're considered to be in renal failure. That is related to the blood pressure, but it can also be related to other problems with the kidneys themselves. If the blood pressure drops, and if the systemic blood pressure drops below 70 millimeters of mercury during systole, then there's not enough blood pressure to sustain the GHP, and the GFR will basically go to almost zero, and you won't be producing any urine at all. If the GFR drops by 50% or more, then you will be, producing like 50% or less of the urine that you would normally produce. The problem with that is that you're not able to remove the waste from your blood that you need to remove and excrete in the urine. There's two basic types of renal failure. It can either be acute or it can be chronic. Acute renal failure is in general not as bad, and it's usually caused by something else, like an infection, or it could be, physical damage to the kidneys, just anything that causes a sudden drop in the GFR. But in general, acute renal failure is usually something that you can recover from. So if you had a bad infection, then if that clears up, then your kidney function would recover. Chronic renal failure is caused by, well, different things can cause it, but most cases of chronic renal failure are caused by diabetes or hypertension. Both of those conditions cause damage to delicate blood vessels. Hypertension damages delicate vessels because you have too much force exerted on the walls, and those capillaries only have simple squamous forming their walls, so they really can't withstand that much force. In diabetes, it's a little bit different, even though, well, I guess diabetes and hypertension frequently do go together, but in diabetes you have high, if it's not controlled, then you will have high blood sugar, and all of that extra sugar that cannot be taken up by your cells is going to end up being metabolized right there in the blood, and that creates toxic products, the reactive oxygen species, that are damaging to cells and cause a lot of damage to capillaries throughout the body. So in both of those conditions, you have damage to all of your delicate capillaries, and some of the most prominent damage is occurring in the kidneys. Chronic renal failure is, in general, much more serious than acute. You generally won't notice it for a long time as it's developing until it gets pretty bad. But because it's been such a long time that you've been having this damage to the capillaries, like to the glomerulus, it's pretty hard to come back from that. And of course it can be deadly if you lose the ability to filter waste from your blood. Uremia is a condition where you have these nitrogenous wastes building up in the blood because of the renal failure, because your GFR is too low to excrete enough of them as urine. Symptoms of uremia include fatigue, and then if it progresses, you can enter a coma and die. This actually had been happening to my mother-in-law. She had renal failure, partly probably due to her diabetes, and also due to her heart failure and some of the drugs she was taking for that heart failure. And it was she was just really tired for several days, and just very tired and sleeping a lot, but also very tired when she was not asleep and kind of nodding out almost a lot. And didn't really understand why. My husband actually took her to lunch and hung out with her after she had been this way for a few days, and he was just on her the whole time to go to the hospital. She hated going to the hospital. She never wanted to go, but eventually he was able to convince her to go to the hospital. And at the hospital they discovered that her kidneys were essentially, her GFR was essentially almost zero, and she had a lot of waste building up in her blood, and that's why That's why she was so tired and unable to sustain her consciousness. In her case, they were able to get her enough GFR to sustain herself without dialysis, which she was very, very happy about. If you're not able to fix the renal failure, at least to have enough of a GFR to remove, enough waste that you won't have uremia. If you're not able to do that, then you'll have to go on to dialysis. Dialysis is kind of the artificial removal of some of those toxic waste from the blood when the kidneys can't do it. You have two basic types of dialysis. Hemodialysis is where you actually filter the blood through a big machine. So that would be like this, where the person's blood is actually removed and filtered through this machine and then pumped back into their body. And then the machine is actually removing the waste and making sure that the contents of the blood are appropriate. So that's the more extreme form of dialysis. If your renal failure is a bit less bad than that, then you may be able to do the peritoneal dialysis, where wastes are removed from the peritoneal cavity, and that's something where you don't have to go to a clinic. You could actually do it at your house. So in peritoneal dialysis, the idea is that you have, your blood vessels, and a lot of those wastes that are in your blood would actually be exiting the blood vessels in the abdominal pelvic cavity and winding up in the fluid in the peritoneum, or the peritoneal cavity, which is the big cavity in your abdomen that's formed by the folds of the largest serous membrane, the peritoneum. In the peritoneal dialysis, you actually have a port inserted into your abdomen, so you can get a catheter in through there, and you're basically pouring in to your peritoneum this solution for the dialysis, and it mixes with the waste, and the waste all solubilize in that solution, and then you pump it back out into this little waste bag that you would then throw away. So that's, a lot easier and actually safer than the hemodialysis, but it doesn't work as well, of course, because you're not removing the waste directly from the blood. Renal clearance is a way of estimating the GFR. It's really hard to measure the GFR directly in, a person who's like alive and well, because you would need to kind of get up in there. So renal clearance is kind of a way of estimating what the GFR would be based on observing how much of a waste product is in the blood versus how much comes out in the urine. So it's a way of estimating or I guess measuring how fast a substance is removed from the blood, and then that is used to estimate the GFR. In order to have the renal clearance actually reflect the GFR, you have to be careful about what substance you're measuring, because some substances are also subject to tubular reabsorption and or tubular secretion, which would mean that they are filtered in the glomerule, in the renal corpuscle. They're filtered into the filtrate, and then in the tubules, they're either put back into the blood, or more of the waste product or of the substance that is in the blood is taken from those peritubular capillaries and put into the filtrate in the tubules. So those processes will kind of distort the renal clearance and make it so that it doesn't really reflect the GFR exactly. So ideally you want something that is filtered in the corpuscle but is not reabsorbed or secreted in the tubules. The two most common things that are used for this, you could actually use, a lot of different substances, but usually they use creatinine, and if they need something more accurate, they would use inulin. Creatinine, we had mentioned, it's generated by your muscles. That's the main source of creatinine in the body. The muscles would be using creatine phosphate as a source of ATP when they're contracting, and then that gives you creatine after you've transferred the phosphate group to ADP to make ATP, and then that creatine is converted to creatinine to be excreted as a waste product. And that would be mostly excreted in the urine because it's water soluble. So if you're doing a creatinine clearance test, then you're looking at how much creatinine is in the blood, and comparing that to how much creatinine is in the urine. And so you can kind of figure out, there's, I guess, some basic values for how much creatinine should be in the blood based on your gender, your age, your size, your muscle mass. The main thing that affects it is how much muscle mass you have. So you have to know basically how much creatinine should be in the blood, and then you can know like how much is coming out in the urine, and that way you can figure out how quickly are the kidneys removing it from the blood. Creatinine is actually subject to secretion in the tubules. So most of the creatinine that ends up in the urine is getting there in the corpuscle. It's coming by being filtered through the glomerulus. But you also have a little bit that is secreted by the tubules. And of course that can also vary kind of from person to person and from day to day actually. So that makes it a little bit less accurate. Also, the fact that how much creatinine you have in your blood is going to depend on your muscle mass makes it less accurate because it's hard to have reference values that are going to be the same for everybody. But what's good about the creatinine test is that it's just really easy. You'll always have creatinine in your blood. Yeah, and that just makes it a really convenient and easy test to do. So that's the most common test that they'll do for renal clearance. Another thing to note about that test is that if you have an extremely abnormal result, it might indicate, I mean, usually that would indicate that something's wrong with the kidneys, but it could also, in some cases, indicate muscle damage. If you have a lot of muscle damage, then that degradation of the muscle cells can cause them to release a lot more creatinine than they otherwise would, which would throw the results of the test off. If you need a more accurate test than that, then they would usually do the inulin clearance test. Inulin is a polysaccharide that is found in certain types of plants. So that's not something that you would normally have in your body at all. That makes it a more difficult test because you have to inject it first. So in the inulin clearance test, you inject inulin into the blood, and it's actually not just a bolus injection where you're doing, one injection, but you kind of have to hook the person up to an inulin drip, so they're getting a constant rate of inulin added to their blood. So they would have a constant level of the inulin in their blood, and then you measure that, and then you measure how much inulin is coming out in their urine. So that's a much more complicated test to do, but it is very accurate because inulin is not at all reabsorbed or secreted by the tubules, so all of the inulin that is in the urine got there from the glomerulus by glomerular filtration, and so that truly reflects the glomerular filtration rate. So now we'll talk about the other two processes involved with the formation of urine, which are tubular reabsorption and tubular secretion. We're going to talk about them together because they involve a lot of similar processes. First, we'll talk about why this even happens. For reabsorption, most of the filtrate that you form initially in the corpuscle is actually reabsorbed. About 99% of the water and about 99% of the solutes that are not waste products would be reabsorbed back into the blood, into the peritubular capillaries from the tubules. So why even make all of that filtrate in the first place if you're just going to be reabsorbing all of it, almost all of it? Well, the answer is because you need to get rid of those waste products very quickly. They need to be kept at quite a low level in the blood, and in order to do that, you have to have a high GFR. You have to be getting like pretty much all the stuff that you can out of the blood and into the filtrate, and then anything that you actually needed to have in the blood, then you just take it back by reabsorption. So that's one of the reasons why you're doing reabsorption. Then for secretion, another reason why you need that is because there's some waste products that actually won't make it past the filtration membrane in the renal corpuscle, so they can't be filtered into the filtrate that way. They have to be specifically added to the filtrate by tubular secretion. And then both of those processes, reabsorption and secretion, allow you to have a very fine level of control over the concentration of the urine and exactly what's in it. So you can have just the right amount of water in the urine, which means you would have just the right amount of water in your blood. And for all the different electrolytes, you can adjust their levels so that you have the right amount of ions in your blood, and whatever's extra would be put into the urine. If you were just using glomerular filtration, that's not possible to do because you're just filtering out anything that's small enough to make it through the filtration membrane without regard for what it is or whether you need it or not. In tubular reabsorption and secretion, you can actually specifically select, you can select specific things to put them back into the blood or put them into the filtrate. So we'll go through the methods by which these substances would be moving either into the filtrate or out of the filtrate in the tubules. In general, you can either be going through the cell or around the cells if you're a substance trying to get in or out of the filtrate. If you're going by the paracellular route, that means that you're passing between the epithelial cells of the tubules. If you're going by the transcellular route, that means that you're passing through the epithelial cells of the tubules, and that one's a bit more complicated. All of these substances that are moving in and out of the filtrate are going to be leaving the lumen of the tubule, the inside part of the tubule. Initially, they're going to end up in the interstitial space, and then they're going to end up in the blood, either in a paratubular capillary or in a vasorectal capillary, depending on which type of nephron we're talking about. So if I look on the diagram, right, so here's the lumen. This would be the lumen of the tubule. So stuff is coming out from there. It's going across. It could either go between the cells paracellularly, or it could go across the cell directly. That's transcellularly. Then it's going to end up in the interstitial fluid. Then it's going to end up in the capillary. That's for reabsorption. For secretion, of course, it would just be the opposite direction. In the paracellular route, everything that's passing between the cells is doing so passively, which means that you're not using any ATP, you're not using any energy, and everything is going to be moving down a concentration gradient from the side where you have more of it to the side where you have less. Unless we're talking about water, in which case it would be moving by osmosis, which is also a passive process in which the water would be moving to whichever side has the most solutes. And in general, the water will be moving to whichever side has the most sodium. It follows the sodium. In the paracellular route, you can't control what is going across or how much. Anything that can fit between the cells of the tubular epithelium is able to cross by the paracellular route. You do have some tight junctions between those cells, but they have gaps in between them so that stuff can go through. And you really can't control how much of anything that's going through is going to go through. How much goes through is just going to depend on the concentration gradient for it. Only small things can go through between the cells. That would be water and small ions. Larger things like glucose cannot go between the cells. That has to go by the transcellular route. So for the transcellular route, that you can also have water and ions going by the transcellular route, and you can also have your bigger things like glucose, amino acids, waste products, and different things like that. Creatinine would be going transcellularly, so bigger stuff. The transcellular route can be passive, so you can have osmosis, and you can have osmosis, and you can have passive diffusion. But you can also have active transport, which would cost ATP, and in which the substances would be moved up their concentration gradient, not down. And in the transcellular route, the cells have the ability to control how much of everything is crossing and what specifically is crossing by controlling the little channels that would let it be crossing. And we'll go through, there's different types of transcellular transport, and we'll go through each of them. Most of the transport to get into the endothelium itself is passive. You have very little active transport in or out of the endothelium. And we're mostly going to be focusing on transport across the tubular epithelium and not really worrying about transport across the endothelium of the capillaries. So for the transcellular transport, it can either be passive or active. In passive transport, you do not need energy, so it does not cost ATP, and something is always moving down a concentration gradient. Those two things will hold true for all types of passive transport. And you have two basic types of passive transport. First, you have simple diffusion. In simple diffusion, something that can cross the membrane is just going across the membrane down its concentration gradient. That only applies for things that are able to cross the membrane by themselves. So they must be permeable to the, or the membrane must be permeable to them. They must be permeable substances. So that would include nonpolar, small nonpolar things. Those are the most permeable things. Anything that has a charge is not permeable because the inside of the membrane is it's fatty, it's hydrophobic. Anything that has a charge is gonna be repelled. It's not gonna be able to cross by simple diffusion. Nonpolar things are also hydrophobic, so they can cross easily across the hydrophobic membrane. Large things have trouble getting across. Small things get across more easily unless they have a charge. In facilitated diffusion, you have the ability to control what's crossing better because the cell is actually going to be putting a little path for it to take. We're talking about something that is impermeable. It does not go across the membrane by itself, and so if it's going to cross, the cell needs to make a little path for it to cross by. And the cell is going to do that using a protein, either a channel protein. We talked about channel proteins a lot in AMP1 when we were talking about the nervous system. So you have ion channels that let different ions cross through. You could also have a carrier protein, which would be for bigger things that are bigger than an ion, like maybe glucose, where it actually is open on one end, it picks the item up, it undergoes a conformational change to be open on the other end, and it drops the item off. You don't need to know the details of that, but both of these would be types of facilitated diffusion, which means that the cell is not spending any energy to drive it, it's free, and the substances will always move from where you have more of it to where you have less of it. That's what makes it count as diffusion. Even with the carrier protein, where the protein is undergoing a conformational change, it's still just diffusion because you're always moving from where there's more to where there is less. So this would be facilitated diffusion here, and then this here is simple diffusion, where you have a substance permeable to the membrane just crossing over it by diffusion. Apart from passive transport, you have active transport. In active transport, you are using ATP, so it costs energy, and you're always moving something up the concentration gradient. So you're taking it from the side where there is less and putting it on the side where there is more, and doing that always costs energy, which makes sense if you think about putting a bag of tea into a glass of hot water. Of course, if you just wait, the tea molecules will diffuse out of the bag into the glass of hot water, and eventually you will have tea. But if you start with tea and you want to put the tea back in the bag, that's going to be hard to do. That's going to cost some energy. There's two basic types of active transport. Active transport itself, which could also be called primary active transport, and then there's also secondary active transport, which is also called co-transport. So primary and secondary active transport, but primary active transport is also usually just called active transport. In that primary active transport, the regular type, you would have a pump protein like this. It takes the substance from where on the side, it picks it up on the side where you have less of it, and then it uses ATP, it undergoes a conformational change, and it drops it off on the side where you have more. So it's pumping it from the side with less to the side that has more, and it costs ATP. The sodium-potassium pump is the most famous example of that. Then in secondary active transport, also called co-transport, the protein that is letting the substance cross actually is not using ATP directly. ATP would be used indirectly. This figure up here is showing secondary active transport. On this side, you actually have that sodium-potassium pump. The sodium-potassium pump is a regular primary active transport pump. It's taking sodium from the side where you have less, which is inside the cell, and putting it on the side where you have more, which is outside the cell. At the same time, it's taking potassium from the side where you have less, which is outside the cell, and putting it on the side where you have more, which is inside the cell. And that process requires ATP. So this sodium-potassium pump is operating all the time in all of your cells and using a significant amount of your ATP. And one of the things that it's doing is establishing and maintaining those concentration gradients for sodium and potassium, so that there's always going to be more sodium outside of the cell and very little inside of the cell. Then that concentration gradient that has been set up for sodium can be exploited by secondary active transport to move something else. So in secondary active transport, you have a protein called a cotransporter protein, and it is going to allow the sodium to flow down its concentration gradient, and it doesn't have to be sodium, but it's going to allow something to flow down its concentration gradient, and then it's going to use the energy of that movement to pump something else up against its concentration gradient, in this case glucose. So this particular cotransporter is allowing sodium to flow down its concentration gradient into the cell, and it's using the energy of the movement of the sodium to move glucose up its concentration gradient into the cell from where there is less to the side where there is more. This cotransporter does not use ATP, but it does have to have the sodium flowing down the concentration gradient. In order to get that concentration gradient for sodium pointing the right way so that you have more on the outside and less on the inside, you need to be running the sodium-potassium pump, and that does cost ATP. So that is how ATP is being used indirectly in secondary active transport or cotransport. You have two types of cotransporter proteins, antiporters and symporters. In an antiporter, the two substances would be going in opposite directions. In a symporter, the two substances would be going in the same direction. So in that example that we have for active or for secondary active transport, where sodium is going down its concentration gradient and glucose is being pumped up its concentration gradient, that is a symporter, because both of the things are moving in the same direction. They're both moving from the outside of the cell to the inside of the cell, but of course one of them is moving down its concentration gradient and the other one is moving up. So always one thing will be moving down its concentration gradient, the other thing will be moving up. A symporter would be putting both of those things into the cell. An antiporter would be putting one of them out and the other one in. So first we'll talk about reabsorption in the proximal tubule. Most of the reabsorption that is carried out by the kidneys is occurring in the proximal tubule. And that's largely because in the proximal tubule, that's where you have microvilli in the epithelium. So the epithelium in the rest of the tubules does not have the microvilli. Those microvilli are increasing the surface area for reabsorption by a lot, by a massive amount. And that lets you get a lot of reabsorption done. Some of that, a good amount of that reabsorption is active. And that requires ATP. So the proximal tubule has a very high requirement for ATP. Out of the whole nephron, it is requiring the most ATP. A bunch of different things are being reabsorbed in the proximal tubule, which makes sense since it's reabsorbing most of the stuff. So a bunch of different electrolytes, so sodium, chloride, potassium, those are all reabsorbed there, but also other things like sulfate and phosphate, and even more different electrolytes like even magnesium and stuff would be reabsorbed in the proximal tubule. Also bigger things that would be counting as nutrients, that would include glucose, amino acids, vitamins, anything that is small enough to make it into the filtrate but that you actually need to have in your blood, you're going to reabsorb it because it's a nutrient. Anything like that, you would reabsorb it in the proximal tubule. You're also reabsorbing a lot of bicarbonate, and of course water. So whenever we're talking about the epithelium of a tubule, you always have an apical side and a basal side. The apical side is going to be the side that is facing the lumen of the tubule, so it's facing towards the interior space of the tubule where the filtrate is, and that's the side that has the microvilli. Then the basal side is the opposite side. It would be facing out. It's the superficial side, so that's facing the external side of that tubule. And the paratubular capillaries would be nearby to that. Sodium is probably the most important substance that is reabsorbed in the proximal tubule because the reabsorption of sodium is used to drive the reabsorption of many of the other things that are reabsorbed there in the proximal tubule. So we'll talk more about that. You have several different ways for sodium to be reabsorbed in the proximal tubule. First, you have on the apical side leak channels for sodium. A leak channel is a channel that will let sodium pass through by facilitated diffusion and is sometimes active and sometimes not active. So it just kind of opens and closes at random. Then on the basal side of the proximal tubule, you have the sodium-potassium pump. So in this image here, we're actually not showing the leak channels for sodium, but right here we do have that sodium-potassium pump. So sodium would be entering from the filtrate into the tubule cell through that leak channel, and then on the other side is going to get pumped out by the sodium-potassium pump in a process that requires ATP. So with this setup, you're going to have a lot of sodium in the filtrate and a lot of sodium in the interstitial fluid, but not a lot of sodium inside of the cell, because as soon as sodium leaks into the cell, you're pumping it back out into the interstitial fluid on the outside of that tubule. From there, it would enter into the blood. Those sodium concentration gradients are going to be really important. They're used by secondary active transport to reabsorb a bunch of different things, different ions, glucose, amino acids, and a bunch of different things. So here it's actually showing sodium being used to reabsorb glucose, sodium being used to reabsorb amino acids, sodium being used to reabsorb ions like phosphate, citrate, and lactate. And there would also be others. Sodium is used to reabsorb a lot of stuff there. So these are all symporters. These are all secondary active transport co-transporters that are transporting both substances in the same direction, so it's a symporter. So for this one, if we zoom in on the sodium glucose one, sodium is moving down its concentration gradient from the side where you have more, the filtrate, to the side where you have less, the interior of the tubule cell. The energy from sodium moving down its gradient like that is used to pump glucose into the cell up its concentration gradient. So you would actually have less glucose in the filtrate and more inside the cell, and you're pumping it up into the cell using the energy of sodium flowing down its gradient. And the same situation applies for this symporter, bringing in amino acids, and for this symporter, bringing in phosphate, citrate, or lactate. That concentration gradient is maintained, in this case, by the sodium-potassium pump on the basal side of the cell. So as soon as you get sodium rushing into the cell through these symporters and through the leak channels, you're going to get sodium pumped back out by the sodium-potassium pump. So that way you always make sure you have very low sodium inside the cell. As long as you have low sodium inside the cell, you're going to have sodium flowing down its concentration gradient into the cell, and you're going to have this co-transport active. For all those things that are being reabsorbed on the apical side using symporters, those are going to be put out on the other side into the interstitial fluid using facilitated diffusion, just carriers for those different things. So they end up having a higher concentration inside the cell and a lower concentration out in this interstitial fluid. So to get them out, it's very simple. You just have a carrier protein that lets them flow down their concentration gradient out of the cell. Also, the sodium is being used to secrete hydrogen. So we're supposed to mostly be talking about reabsorption right now, but the sodium, you have an antiporter here that's actually allowing sodium to flow down its concentration gradient into the cell, and then it's using the energy of that movement to pump hydrogen ions up their concentration gradient into the filtrate. So the cell would have more hydrogen ions, and then the filtrate has less. So that's a secondary active transport antiporter being used for proton secretion. The final thing that's important about the sodium here is that the sodium concentration gradient actually creates a voltage across the cell. So you have more sodium out here in the filtrate, less inside the cell, and then you end up with more sodium out here in the blood. And that creates a voltage across these cells, so where you have a more of a negative charge in the lumen with the filtrate and a slightly more positive charge, or at least less negative charge, in the interstitial fluid on the basal side. And that charge, that voltage, helps chloride ions move. So you would actually have chloride ions passing between these cells paracellularly, and it's going to be moving. Since it has a negative charge, it's going to be attracted towards the side that is more positive. So it's going to be repelled by the negative charge here in the lumen, and it's going to be attracted over to the side that's slightly more positive. And now secretion in the proximal tubule. So as I mentioned, you have hydrogen ions being secreted using the sodium hydrogen antiporter. That relies on the sodium concentration gradients. Then you also have nitrogenous waste being secreted into the filtrate there. Creatinine is secreted into the filtrate. Any water-soluble toxins in the blood that were too big to be filtered initially would be secreted into that filtrate in the proximal tubule. And finally, different drugs. Not all drugs are secreted into the proximal tubule, of course, but a lot of drugs are. Any drug that is very quickly secreted into the proximal tubule, it's going to be eliminated from the body rather quickly, and you're going to have to dose frequently to maintain a steady level of the drug in your blood. So in general, any drug that is quickly eliminated would require more frequent doses. We'll also talk about how bicarbonate is reabsorbed in the proximal tubule because that process is a little complicated. So first it starts with that sodium hydrogen antiporter that is secreting sodium ions into the filtrate. So that means that the concentration of hydrogen ions in the filtrate is high and it's going up. And those hydrogen ions are going to react with bicarbonate in the filtrate, and that forms carbonic acid, H2CO3. Then carbonic anhydrase is going to convert the carbonic acid into water and carbon dioxide. The water can remain there in the filtrate, or it can enter into the cell by reabsorption. The carbon dioxide that's a small nonpolar substance, so that is permeable to the membrane. It's going to just diffuse across the membrane by simple diffusion into this tubule cell. And then on the other side, inside the cell, it's going to recombine with water that happens to be inside the cell. Carbonic anhydrase is going to combine that carbonic anhydrase is going to combine that carbon dioxide with water to make, again, carbonic acid, which immediately is going to dissociate into bicarbonate and hydrogen ions. The hydrogen ion is actually going to be pumped right back out into the filtrate through that sodium hydrogen antiporter. The bicarbonate, on the other hand, is going to leave the cell on the basal side by facilitated diffusion and ultimately enter the blood. And finally, water reabsorption. Most of the water that is being reabsorbed in the proximal tubule is reabsorbed in the second half. That's because in the second half you have had a lot of solutes reabsorbed already, and so the osmotic pressure is changing in a way that favors water leaving the filtrate. If you're in the front part of the proximal tubule, then you have a lot of solutes in the filtrate and comparatively less solutes in the cells and outside in the interstitial fluid. So water is not going to be leaving the filtrate in the first part of the proximal tubule. Osmosis is holding it there. It's always going to stay where you have most of the solutes. Then as you're passing through the proximal tubule, you're having a lot of those solutes being reabsorbed. Sodium is being reabsorbed, and it's taking a bunch of other things with it. So all those solutes are now moving into the tubule cells and out of them into the interstitial fluid and ultimately into the blood. So the solute concentration is rising there in the interstitial fluid while at the same time it's dropping inside the lumen, inside the filtrate. And that condition favors water to move by osmosis out of the filtrate through the cell and into the interstitial fluid and ultimately the blood. That is called obligatory water reabsorption. Because you don't have any choice about whether you do it or not. The body can't control that to make it happen less. It just is going to happen just by osmosis. You have aquaporins there, which are letting the water cross by facilitated diffusion. So these channels, these are actually just channels that are for water specifically. They won't let anything through them except for water. So that's why they're called aquaporins, little water pores. You have some in the apical side, and you also have more on the basal side of the tubule cells. So osmosis is drawing the water from the filtrate through into the cell, and then through into the interstitial fluid, and ultimately into the blood. Here's a zoomed in view of that aquaporin, and here you have a stream of water molecules moving through it. In the center, you have some special amino acids that are keeping any other ions out, or any ions out, so only water can go through there. Water is able to cross the membrane a little bit. It is polar, so it doesn't really like to be inside of the cell membrane because that's hydrophobic, and water is obviously hydrophilic. But it is also very, very small, so it can get through a little bit. But you're not going to have water getting through fast enough for it to really mean much by simple diffusion. So in general, when water is going to cross a membrane in any meaningful way, it needs to have those aquaporins there so it can cross by facilitated diffusion and osmosis. So as this process continues, as you're moving through the latter part of the proximal tubule and water is being reabsorbed this way, the filtrate is again becoming more and more concentrated. So the concentration of the filtrate is actually going to vary throughout the whole tubule system, and it gets a little complicated. It starts out being pretty concentrated, then you have reabsorption of solutes in the first part of the proximal tubule, so it gets less concentrated. Then water starts moving out by osmosis, so then it starts getting more concentrated in the latter part, in the very last part of the proximal tubule. As that filtrate becomes more concentrated because the water has been reabsorbed by osmosis, now the concentration gradients are favoring the reabsorption of more solutes. So then you have more solutes being reabsorbed. That causes the filtrate to become less concentrated. Now osmosis is favored again. You have more obligatory water reabsorption. The filtrate becomes more concentrated again. It's just a process that repeats. So after the proximal tubule, we have the nephron loop, which is also called the loop of Henle. You have had most of your reabsorption occur already in the proximal tubule, especially stuff like glucose and amino acids and vitamins. They've basically all been reabsorbed already. You still have some water that's going to be reabsorbed and some electrolytes to be reabsorbed. Everything else, basically the reabsorption is already done. So you have some more of that water and electrolyte reabsorption occurring in the nephron loop, as well as in the distal tubule also. The nephron loop is actually really important for adjusting the concentration of the urine, but we're going to talk about exactly how that happens later on after we get through tubular reabsorption and secretion. So the reabsorption of water and electrolytes differs between the descending and ascending limbs of the nephron loop, and that's because in the descending limb it is water permeable, meaning you have plenty of aquaporins in the tubule epithelium cells, but it's not permeable to the electrolytes. So that means you don't have channels that would allow those ions to cross through the cells. So you do have aquaporins, you do not have ion channels in the descending limb to any significant degree. So in the descending limb, water is going to be leaving by osmosis. You've already had a bunch of the solutes filtered out of the filtrate, or reabsorbed out of the filtrate already. So that means that the solute concentration of the filtrate has dropped significantly by the time you get to the end of the proximal tubule. That means that conditions favor water to be reabsorbed by osmosis. So water is going to be leaving by osmosis and entering the interstitial fluid and ultimately entering into the blood through those aquaporins. The solutes, on the other hand, are not able to leave. Even as the water leaves, the filtrate will become more and more concentrated, and so then the concentration gradients favor the solutes to leave, but there's not channels for them to go out, and so they can't go out. They have to stay in the filtrate. And so as you are going down the descending limb, water leaves and leaves and leaves and leaves, and then the filtrate is becoming more and more concentrated. These numbers here, 300 milliosmoles, 600 milliosmoles, 900 milliosmoles, that's a measure of the solute concentration of the filtrate. So the more milliosmoles you have, the more solute molecules you have in the filtrate. So the filtrate is becoming more and more concentrated as you get to the bottom. It reaches its maximum concentration, which could be up to 1200 milliosmoles. It actually depends on which type of nephron we're talking about, but that's the maximum. Then in the ascending limb of the nephron loop, the situation is basically the opposite. Now you don't have aquaporins in the tubule epithelium cells, but you do have ion channels. So now the electrolytes can cross, but the water cannot get out. So water is going to be trapped here in the filtrate, and the solutes are going to start moving out down by facilitated diffusion through these ion channels. So you have several different types. Well, actually you have many different types, but one of the most important ones would be the sodium-potassium chloride symporter on the apical surface of the cells. That would be bringing all those ions into the cell by facilitated diffusion, all three of them at once. And then on the basal side, you have the sodium-potassium pump that is pumping sodium into the interstitial fluid and pumping the potassium back into the tubule cell itself. So that means that ultimately you're going to get sodium and chloride coming out of the tubule, coming out of the filtrate and entering interstitial fluid and ultimately the blood. But the potassium is not going to come out really. It is going to be removed from the filtrate and go into the tubule cells, but it's going to get caught up there because as soon as it goes out on the other side, the sodium-potassium pump is just going to put it right back inside the tubule cell. So then as you go up, you're losing those solutes, prominently the sodium and chloride from the filtrate. And so you're reducing the concentration of the filtrate. The concentration is going to drop lower and lower the higher you go in the ascending limb. And then we come to the distal tubule. So by this point, virtually all of the water and electrolytes that you are going to reabsorb have already been reabsorbed. But actually the little bit that you have left to reabsorb, you have to reabsorb that last little bit. If you don't do it, you're going to be losing again multiple times your own blood volume every day in urine, which is not sustainable. So you have to reclaim that last little bit in the distal tubule, and a little bit actually in the collecting duct. So now you have what's called facultative water reabsorption. So up until now, all the water reabsorption we've been talking about has been obligatory. There hasn't been a choice about whether you do it or not. But in the distal tubule, you have facultative water reabsorption, which means that the cells can control how much water is reabsorbed. So here we have hormones acting to retain water or to excrete water. So the distal tubule is the site for most of those hormones that are affecting the urine contents to act. So you have aldosterone acting there, ADH acting there, and AMP and BNP acting there. Aldosterone is going to be increasing the amount of sodium-potassium pumps that you have, the sodium chloride symporters, the sodium channels, and the hydrogen pumps. So you can see a lot of that in the diagram here. This is the lumen side that has the filtrate. On the other side is the blood side, so you would actually have interstitial fluid there, but frequently it's kind of left out of the diagrams. And then just next to that you would have the paratubular capillaries with the blood. And then these, of course, are the cuboidal cells forming the epithelium of the tubule. So here's the sodium chloride symporter. That is bringing sodium and chloride both into the cell by secondary transport. And then that sodium is going to be pumped out of the tubule cell into the interstitial fluid and ultimately the blood by the sodium potassium pump right here. You're actually having potassium being pumped back in, and then it's recycled. You have a channel that lets it right back out, so you can keep running this pump. And then here's your sodium channel. So you also have sodium channels on the apical side that are allowing sodium to flow in flow into the tubule cells, and then they would be pumped out. The sodium would be pumped out by the sodium-potassium pump. And the hydrogen pumps are actually not shown here, but those hydrogen pumps would actually be going the opposite direction. They would be secreting into the filtrate on the apical side. So all of these pumps are causing sodium to be reabsorbed, chloride to be reabsorbed, hydrogen to be secreted, and potassium actually to be secreted ultimately, because you can't get rid of it into the blood. As soon as you do, you're taking it back up through the sodium-potassium pump. And so that is how aldosterone is doing all of its actions, to increase retention of sodium and chloride and to increase excretion of potassium and hydrogen. And then water would ultimately end up following the sodium out by osmosis. ADH is increasing aquaporins, so it's causing aquaporins to be inserted into the apical and basal membranes. If you don't have ADH present, ADH again is antidiuretic hormone, as you remember from AMP1. That's one of the main hormones that is responsible for retaining water and stimulating thirst, to maintain your blood volume and your blood pressure. And ADH's main, well, apart from its actions in the brain to stimulate thirst, ADH's main action is in the kidneys, and it inserts aquaporins into the membranes of these tubule cells in the distal tubule. If you don't have the ADH present, you're not going to have very many aquaporins in the membrane at all, and it will not be very permeable to water. So water wouldn't be able to cross. So aldosterone increases water retention because water follows the sodium, but only if ADH is present. You have to have the ADH there, and that gives you aquaporins, and then water has a pathway that it can take across the cells to get out of the filtrate by osmosis. Otherwise, it does not have a path. Then you also have AMP and BMP acting there in the distal tubule. They are actually decreasing the amount of sodium-potassium pumps that you have in the membrane, and they're decreasing the amount of sodium channels that you have in the membrane. So that means that you're not going to have as much sodium being reabsorbed. The sodium is going to be excreted in the urine instead, and water in that case would end up following the sodium there. And of course, AMP and BNP also inhibit the release of renin, and they also actually inhibit the release of ADH. So those hormones are always kind of antagonistic to each other. So that if you have AMP, that's going to prevent you from having as much ADH and aldosterone by preventing the renin. You also have some reabsorption and secretion in the collecting duct, and it's basically the same as the processes that you have occurring in the distal tubule. So you have the same hormones acting there, you have the same channels, and the same processes where your sodium and the chloride are being reabsorbed. Water is being reabsorbed if you have ADH present. Hydrogen ions are being secreted. And if you have ANP, then water would be secreted instead of reabsorbed. And sodium would also be not reabsorbed, so it would end up being eliminated in the urine. You also have a reabsorption of urea in the collecting duct and actually in the distal tubule as well. I just didn't mention it, but here is a collecting duct cross section, so you can see sodium and chloride are being reabsorbed there. Water is being reabsorbed, but only if you have ADH. Hydrogen ions are being secreted. Urea is being reabsorbed, and actually bicarbonate would be reabsorbed as well. The urea is a waste product, so it probably seems weird that you're reabsorbing it there. Ultimately, you're not going to be keeping that urea. You will get rid of it, and how you're going to do that exactly, we will talk about when we're talking about how urine gets concentrated. So here's kind of a summary of the reabsorption and secretion in the tubules. In the proximal tubule is where we have most of our reabsorption occurring. You're absorbing over half of the water. pretty much all of the glucose, amino acids, and other nutrients like vitamins and such. You're also reabsorbing most of your bicarbonate and most of your electrolytes like sodium, potassium, chloride, magnesium, phosphate, sulfate. Most of that stuff is all getting reabsorbed in the proximal tubule. You're also secreting stuff. You're secreting hydrogen, you're secreting nitrogenous waste, and other toxins that are water soluble, and certain drugs that are water soluble and subject to secretion in the tubules. Just to remind you, the water reabsorption that you have occurring here in the proximal tubule is obligatory water reabsorption. So there's no way for the body to adjust how much water is reabsorbed. It's just happening by osmosis. Then in the nephron loop, in the descending limb, you have water being reabsorbed. Another significant portion of the water is reabsorbed there, and because you have aquaporins there, but you do not have ion channels there, so no ions are leaving. So it's becoming more and more concentrated as it goes down, and water leaves, but ions do not. Then on the ascending limb, the opposite situation occurs. Now you don't have any aquaporins, but you do have ion channels. So now the solutes start leaving. Mostly sodium and chloride would be reabsorbed there in the the ascending limb of the nephron loop, and the water does not go anywhere, it stays in there. So now the filtrate is going to become less and less concentrated as it's coming back up, until it gets back to the top and enters the distal tubule. At that point you have the reabsorption of the last little bit of water that you needed to get back to maintain your life. Also reabsorption of that last bit of sodium and chloride that you needed, and you're secreting potassium and hydrogen as well. And to remind you that the water reabsorption that you have there is facultative water reabsorption, which means that the body can control how much of it is being reabsorbed using different hormones like ADH and aldosterone. Well, yeah, ADH and aldosterone and ANP, BNP. Then in the collecting duct, the same situation applies basically as what you had in the distal tubule. You are reabsorbing water facultatively. You're reabsorbing the last teensy bit of sodium and chloride, and you're secreting hydrogen ions, and you also would actually be reabsorbing urea in the distal and collecting duct as well, in order to concentrate the urine. And so your hormones that are acting on the nephron to adjust the contents of the water or of the urine are all acting here in the distal tubule and the collecting duct, although the collecting duct is technically not part of the nephron. And so here you have facultative water reabsorption that is always dependent on the presence of ADH to insert the aquaporins into the membrane. Oh, right, and then your filtrate would be, moving down the collecting duct, and it would get to the end. At that point, it's going to enter the minor calyx, and it's now considered urine, actually. So now we'll talk about how the body is controlling the water content of your urine or the concentration of your urine. So in order to form dilute urine that has a lot of water in it, this is what you would do. Dilute urine is hypotonic, which means it has less solutes in it than the blood. That means that it has less osmolarity than the blood, and its osmolarity would be less than 300 milliosmoles. So 3 milliosmoles, again, is the way that you're measuring overall solute concentration. It's actually measuring the moles of all solutes that are present in a solution, and a mole is a way of just measuring how many actual molecules of solute are there. So the milliosmoles is measuring how many actual molecules of solute are present in a solution of all types of solute. And that is determining the osmotic pressure that encourages water to enter or leave a compartment by osmosis. The blood is, if things are correct, then the blood should be about 300 milliosmoles in its concentration. So in order for urine to be dilute and be hypotonic, it needs to have less than that. It needs to have less osmolarity than 300 milliosmoles and less solutes than the blood has. This would occur if you don't have very much facultative water reabsorption. So anytime you don't have ADH, basically. If you don't have ADH, then you're not going to have aquaporins in the distal tubule or in the collecting duct, and you're not going to be reabsorbing water by osmosis in those areas. And that gives you very dilute urine because you are still having, especially because you are still having solutes being reabsorbed, just not water. So you start out with filtrate that is essentially the same concentration as the blood in terms of its solutes. It's around 300 milliosmoles. As you go through the proximal tubule, the concentration of solutes does kind of rise and fall depending on where osmosis is occurring, kind of cyclically, but overall you're hovering around 300 milliosmoles. And then you start going into the descending limb of the nephron loop. As you do that, the solutes stop leaving, but the water continues to leave, which means that the filtrate is becoming more and more concentrated until you get to the bottom. It could be 900 to 1200 milliosmoles by the time you get to the bottom, so 3 to 4 times as concentrated as the blood. Then when you go to the ascending limb, now you don't have any aquaporins, so now water is trapped in there, and the solutes are going to start leaving. And as they leave, you actually end up having a lot of solutes leave, so you end up with urine that's around 150 to 100 milliosmoles when it's coming out of the nephron loop and entering the distal tubule. At that point, if you don't have more water leaving, but you do have more solutes being reabsorbed, the concentration will continue to drop. And it can get down to around 50 milliosmoles, which would be 1/6 as concentrated as the blood. So very, very dilute. That relies on not having aquaporins. If you have aquaporins in there because you have ADH, then you're not going to make dilute urine. So the only way to make that dilute urine is if you do not have ADH released. And so ANP inhibits the release of ADH, which it needs to do. If it doesn't do that, then it won't be able to cause you to eliminate water. Now we'll talk about concentrated urine formation, which is more complicated. So concentrated urine is the opposite. It's hypertonic. It has more solutes in the blood, more osmolarity than the blood. So that means that its osmolarity has to be greater than 300 milliosmoles, which is how much solutes you have in the blood. The concentrated urine could be anywhere between just above 300 milliosmoles up to 1200 milliosmoles. 1200 milliosmoles is the maximum concentration that you can form. That would be 4 times more concentrated than the blood. If you have a lot of facultative water reabsorption in the distal and collecting duct, then you're going to end up with concentrated urine, and that requires ADH. So if you have ADH, then you're going to have aquaporins in the cells lining the distal tubule and the collecting duct, and that means you're going to have water leaving by osmosis. As more and more water leaves, the urine becomes more and more, or the filtrate becomes more and more concentrated until you get to your final product, up to 1200 milliosmoles concentration in that urine. Very little water. So not only do you have to have ADH to do this, but you also need to have the interstitial fluid here more concentrated than the filtrate. So if you have 1100 milliosmoles here in your filtrate, you have to have more than that. You have to have more solutes than that in the interstitial fluid. Otherwise, water will never leave through those aquaporins. It'll stay. Right, so you have to have more solutes outside of the collecting duct if you're going to have water leaving by osmosis. You have to have a path for it to go, and you also have to have a reason for it to go. It has to be attracted to more solutes outside than in. And so the rest of the time, or the rest of the presentation, we're going to talk about how you're creating that interstitial fluid that is so concentrated that you can get water still leaving the filtrate, even though the filtrate itself is already very concentrated. So the mechanism that achieves that is the countercurrent mechanism, and you find that in the juxtamedullary nephrons. So the juxtamedullary nephrons are making sure that the interstitial fluid in the medulla is more concentrated than the urine itself or the filtrate itself in the collecting ducts, and then every nephron, including the cortical ones, will benefit from that and be able to make concentrated urine. The countercurrent mechanism has 3 different parts of it. It has the countercurrent multiplier, it has urea recycling, and it has the countercurrent exchanger. So we'll go through each of these in turn. The countercurrent multiplier and the urea recycling together are making that high solute concentration in the medulla. The countercurrent exchanger is making sure that the solute concentration is maintained. If you didn't have that, then all those extra solutes that you end up with in the medulla would just enter the capillaries and be carried away by the blood. So you have to have the countercurrent exchanger as well. The solute concentration in the renal medulla is going to get higher and higher the deeper you go, as established by the countercurrent multiplier and the urea recycling. It can get up to 1200 milliosmoles. It cannot get higher than that. The main solutes that are causing it to be that high are sodium and urea. Well, sodium chloride, so salts, and urea. You have a lot of sodium chloride and a lot of salts in the inner medulla. And you can't get any higher than that, and that's why the urine itself can only be up to 1200 milliosmoles in its concentration. Yeah. So, in this diagram, it's showing this here is the countercurrent exchanger, where you have the blood flowing the opposite direction as the filtrate is flowing through the loop, through the nephron loop. And then the countercurrent multiplier is the water leaving the nephron loop there, and then the salt leaving on the other side. We're going to go through that in more detail. The urea recycling isn't shown there, but you would actually have urea coming from the distal tubule and the collecting duct into the inner medulla. So first, the countercurrent multiplier. This would be occurring in the nephron loop in your juxtamedullary nephrons. It can establish a concentration of sodium chloride in the interstitial fluid of the medulla up to 600 millimolar. Then if you get another 600 millimolar from the urea, you just add those up to get your 1200 milliosmoles. The molar is how many moles of sodium chloride you have there, and the moles are related to the actual number of molecules of sodium chloride that you have. But to figure out the actual number of moles, you have to do a little bit of math, which we're not going to do. So in your ascending limb of the nephron loop, you have sodium chloride exiting into the interstitial fluid. Some of it would be leaving by facilitated diffusion, but the rest of it is actually being pumped out actively. And so that's making the interstitial fluid here, where the nephron loop is, become more and more concentrated with lots of sodium chloride. And then that actually affects the descending limb by drawing water out by osmosis. So in the descending limb, you have aquaporins that allows water to leave, but water isn't going to leave unless the interstitial fluid is more concentrated than the filtrate. And so the interstitial fluid here is more concentrated than the filtrate because of all this sodium chloride that's actually being lost from the filtrate in the ascending limb. You're losing the sodium from the ascending limb so that you can reabsorb water from the descending limb. So here's that water coming out in the descending limb. As you have more and more water exiting in the descending limb by osmosis, what's left behind gets more and more concentrated. So the solutes at the bottom of the nephron loop are super concentrated. And then that concentrated filtrate lets you reabsorb sodium when you get back to the ascending limb. So since this filtrate is super concentrated here, that lets you drive a lot of facilitated diffusion to get rid of that sodium chloride, so that it exits back into the interstitial fluid. And this just kind of works in a loop. You're losing sodium chloride in the ascending limb. That raises the concentration of the interstitial fluid. That lets you lose water from the descending limb, right there. Then this filtrate becomes very concentrated, and that super concentrated filtrate then allows you to lose more water in the ascending limb, or sorry, more solutes in the ascending limb. And that operates in a cycle. And the more times the cycle operates, the more concentrated the interstitial fluid gets in the medulla. And the further down you are, the more concentrated it is. So if you're kind of in the middle, it's going to be moderately concentrated. If you're at the very bottom, it's going to be extremely concentrated, up to 1200 milliosmoles. Then in urea recycling, you're adding urea to the inner medulla, and that also contributes to the osmolarity. So about 600 of those milliosmoles actually would be coming from the sodium chloride, not 1200. The other 600 would be coming from the urea. So when you are reabsorbing water facultatively in the distal tubule and the collecting duct, that means that the urine is becoming more concentrated there, or the filtrate is becoming more concentrated there. And then that allows you to have a concentration gradient that drives facilitated diffusion of urea out of the filtrate in the collecting duct, mostly in the collecting duct, a little bit in the distal tubule, mostly in the collecting duct, and that urea is going to end up in the medulla because the collecting duct is going straight through the medulla. So that gives you a lot of urea here in your inner medulla. As you go further up towards the cortex, you're going to get less and less urea as the concentration of the interstitial fluid drops. But down in the bottom, you're going to have quite a lot of urea, up to 600 millimolar to give you your 1200 milliosmoles of concentration of solutes total. Some of that urea is going to be secreted back into the descending limb of the nephron loop through this transporter, the UTA2 transporter. Some of it would stay in the medulla. A lot of it is actually going to get secreted back into the filtrate, where it's going to go through the tubules again. When it gets to the collecting duct, once again, it will exit by facilitated diffusion and end up back in the inner medulla, contributing to the overall solute concentration there. So it's called urea recycling because you have urea leaving the filtrate, rejoining the filtrate, leaving the filtrate, rejoining the filtrate, over and over in a circle. Some of that urea, of course, is going to be excreted. Actually, the majority of the urea will be excreted. It's quite a lot of urea, but you have so much urea here that even if you're excreting most of it in proper urine, you can still reabsorb a good bit here in the collecting duct so that it can contribute to that solute concentration in the inner medulla. Finally, the countercurrent exchanger, which is responsible for making sure that you don't lose that concentration gradient that you've set up in the medulla interstitial fluid using the countercurrent multiplier and the urea recycling. So if you didn't have this countercurrent exchanger operating, then you would make your inner medulla more concentrated, but then all those extra solutes would just end up being carried away by the blood, and it wouldn't help you form concentrated urine. So this is occurring again only in the juxtamedullary nephrons. Those nephrons have a special blood supply for the nephron loop, the vasa recta. So in the rest of the tubules, you're going to have the paratubular capillaries. If we were talking about a cortical nephron, then the whole tubule system would be surrounded by paratubular capillaries, but in the juxtamedullary nephrons, the nephron loop is surrounded by the vasa recta capillaries, which is different. The vasa recta capillaries have the blood moving in the opposite direction as the filtrate is moving through the nephron loop, and that is really important for the countercurrent exchanger to be operating. So as your vasa recta is entering into the medulla and going deeper into the medulla, it begins to lose water by osmosis. Because this blood is going to start out at 300 milliosmoles, standard blood concentration, and the medulla is getting progressively more and more concentrated as you go down. So even at the very top, it's like 300 milliosmoles, or at the very top of the medulla, it's like 300 milliosmoles. As you go down even just a little bit, you're already at 400 milliosmoles, and you're going all the way down to 1200 milliosmoles at the bottom. So as you go down, the water or the blood is becoming actually less concentrated than the interstitial fluid in the medulla around it, which means that water is going to be drawn out from the blood by osmosis. At the same time, you have, again, more solutes in the interstitial fluid than in the blood, so that sodium chloride is going to be entering the blood by facilitated diffusion. So overall, you're losing water and gaining solutes from the blood, or in the blood. That means the blood is becoming more and more concentrated the deeper you go. At the bottom, the blood will actually be as concentrated as all this fluid around it in the medulla, so it'll be 1200 milliosmoles. As the vasa recta capillary is coming back up the other side, the opposite thing happens. You start to lose sodium chloride from the blood. The blood ends up being a little bit more concentrated than the medulla and interstitial fluid around it, so sodium chloride is going to leave by facilitated diffusion, and water is going to be attracted to all those solutes, so water is going to be entering by osmosis. And now the blood is becoming less and less concentrated as it ascends out of the medulla until it gets back to its original concentration of 300 milliosmoles. So overall, the concentration of the blood is not changing. It starts at 300, it ends at 300, but down at the bottom of the medulla, at the bottom of the nephron loop, it has gotten very concentrated. In this way, any solutes that enter the blood are going back out. So they're not actually being swept away. They're being put back into the inner medulla where they were picked up from. That way they stay ultimately in the inner medulla interstitial fluid. That gives you a very high concentration of solutes in that medulla fluid, and that lets you draw water out of the urine by osmosis so you can make that urine really concentrated. And that's how it happens.